All right, I think we can start because we have a couple of technical notes. Right. I hope everybody can hear me. Hello. It is my great pleasure to welcome you today to a second day of Virtual Symposium 2020. We had some great sessions yesterday, and I'm very excited to see sessions today. If you watched yesterday, you probably know that my name is Karolina Sienkiewicz, and I am a coordinator of Virtual NGSymposium this year. And I'm going to start with a couple of technical notes. First of all, for people that are participating in sessions on Discord, both speakers and organizing team, we had some issues with Discord noises. So I would like to ask you to turn them off. So to do that, you have to go to bottom left corner to the user settings. It's a cog icon. And then when you open the user settings, you have to select notifications and scroll down and turn off the sounds for different parts of Discord. And that would help us to have a greater stream. Also, for speakers that are going to have their talk later on today, our technical team asks you to first join a technical speakers channel, so you are able to set up everything, and maybe we will be able at least half an hour before your talk. So maybe we'll be able to avoid some of the technical troubles that we had yesterday. Regardless of technical troubles, I'm very excited for today's talk, and I hope you are too. We have planned 2 great sessions today, including 2 keynote talks and a summary talk about NGS School Society. So be welcome to join us and listen to them both on YouTube or on Discord. All right. Yes, so this is a schedule for this session, and we're going to start with a keynote lecture by Arro Straton. Arro is an assistant professor at the University of Virginia Public Health Sciences and Center for Public Health Genomics. His research focuses on studying genomic variations and genetic diversity and its consequences on species health and survival. He is a part of Genome Asia 100K consortium. This is a major Asian gene study that focuses on boosting our knowledge about medical genomics, understanding human migration and origins, and decreasing underrepresentation of Asian populations in genetic studies. He's going to tell us more about this in his keynote talk, Mapping Genetic Variations in Asian Populations. Akrosh, welcome. We are very happy that you decided to give this talk. And if you are ready, the floor is yours.
All right, so first, can everybody hear me, and can you see my screen?
We can hear you.
And can you see my PDF or the presentation?
Not yet. Yes, it's a black screen. Can you check if the OBS window didn't crash?
Oh. What about now?
Oh, yes, now we can see it.
Okay. All right. So so you can hear me and my screen is there, so I can start. Is that correct?
Yes.
All right. So thank you, Carolina, for the kind introduction. And I would like to thank the organizers for the opportunity to talk about this project, which I've been involved in for the past few years now. And that is an effort to map the genetic variation in Asian populations. I would like to start with an acknowledgement that all the data, analyses, and results that I will present today has been a result of the efforts of a large number of individuals from several institutions across the globe. So before I start talking about Asian populations per se, I'm sorry, is there an echo suddenly?
There is no echo, but we lost your cam.
I'm sorry, you lost what?
Your camera. So probably the other tab of OBS crashed.
Okay, let me see here.
Now we can see you.
Okay. All right. Can you see my screen again?
It is a bit large, but yes.
Okay. Okay, so
Yes.
So before I start talking about Asian populations per se, I hope we can all agree that doing genomic research in diverse populations has several advantages, and I list some of them on this slide. Right? It can help you identify the genetic influences of health disparities, as seen, for example, in the case of kidney disease, which has a higher prevalence in individuals who have African ancestry. It can help you understand human biology. For example, if you look at double knockouts or homozygous loss of function, it can tell you something about gene function. It can lead to improvements in clinical care. For example, rare truncating variants in PCSK9 are at higher frequency in individuals who are African American. And they have been associated with a protective effect. So they decrease the risk of coronary heart risk. And that means that there are several PCSK9 inhibitors now in trials and under development. However, if the research was only being done in one population, we would have required extremely large sample sizes to come up with the same science. It can also help inform genetic diagnoses. It can tell us about human migration. It can tell us about adaptations. It can tell us about genetic admixture. All of these have shaped both human history, health, and disease. Right? So now, if we agree that genomic research in diverse population is advantageous, we should probably look at what the reality is. So this plot here is from a study done 2 years ago. And what you see is on the x-axis time, and on the y-axis, it tells you how many individuals were involved in a genome-wide association study. So as this plot makes it very clear, most of the individuals who were involved in genome-wide association studies, even up to 2018, are of European ancestry. Now, that is not a surprise, right? The first genome-wide association study was done in 2005, and it was for macular degeneration. And most of the data sets that have come for genome-wide association studies between 2005 to 2018 have been from 3 countries: United States, United Kingdom, and Iceland. So it's not a surprise that most participants in these studies are Europeans. However, if we look at this from the lens of my talk today, about 60% of the world is of Asian descent, and only about 11% of the participants in these studies are Asians. Even out of that, 9% of those participants are of East Asian origin. So in other words, Asia is not at all represented, right? And none of the minorities are. So this was an analysis I did yesterday. And here what you see is not the number of individuals in genome-wide association studies, but the number of studies themselves, right? And you can see the same trends. Most of the studies are done on populations that are of European descent. Again, I'm not saying that this is limited to genome-wide association studies, right? I'm using genome-wide association studies as an example, but this is pervasive. It goes and extends to something as integral as the human reference genome. So as all of us know, the human reference genome is constructed from genetic material from a large number of individuals. And I think all except one of those individuals has a European background. And the result of that is that about 4.6% of the human genome has an Asian haplotype. And for those of you who work with mapping or short read sequencing, you are aware of reference bias and the consequences. So you can imagine how all the results are somewhat biased in certain regions of the genomes. Definitely they are biased. And this in one way reflects disparities in how different ethnicities both interact and benefit from biomedical research, right? But beyond that, if we believe that doing genomic research in different ethnicities has a lot of advantages, this is undermining our efforts as scientists to understand genotype-phenotype relationships, to do proper clinical management, right? To do proper diagnoses in diseases, and so on. And just to bring it home, this is a strong example of the impact of the lack of diversity. So what you see here are these are summary statistics that are derived from a genome-wide association study of European individuals. We are looking at 17 quantitative blood panel traits, and we are using the summary statistics from the European-derived samples in order to predict those traits in different populations. And what you can immediately see is that we are not as successful, right? And you might say, okay, these are blood panel traits. But what you have to understand is this extends. So for example, there is a huge push to understand polygenic risk scores, right? And there we are still going to use summary statistics, and we are still going to use prediction. And the same result will extend to something like polygenic risk scores. And that will directly affect clinical management. So what I'm trying to say is that this is sort of the backdrop in which a few colleagues and some collaborators came together, and we actually decided to form a consortium. So we have, it's a consortium with a large number of individuals from 38 different institutions across the globe. We do have sequencing partners in MedGenome, which is housed in a lot of different countries, including India, and Macrogen, which is, again, a big sequence provider, I think primarily headquartered in Korea. So the goals of this consortium are to sequence individuals in all population isolates within 48 Asian countries. And as I mentioned, one of the problems that we are seeing frequently is the bias within the reference genome. So we are going to generate Asian reference genomes. And because of the scientific interests of the participants of this consortium, there is going to be a focus on oncology, metabolic disorders, and lifestyle diseases. So we've already created or analyzed a pilot data set, which was composed of 1,739 samples from 219 populations. And that publication was featured in Nature in December of 2019, so about end of last year. And a lot of the analyses I'll be talking about today will be from that particular data set. And I will also talk about what we are working on now a little bit in the end. So the pilot data set Panel A here shows the pilot dataset, so the 1,739 samples that we used as part of this dataset. And as you can see, even though the consortium is focused in Asia, we used a lot of samples from around the globe. And that is because you always want to provide context, right? So if you are calculating, let's say, a quantitative measure for a particular population in Asia, it has no meaning till we understand what is the value of that same trait or measure across the globe. And panel B here then focuses on the samples that we actually collected from Asia. And as you will notice, there is a huge emphasis towards India and basically South Asia and Southeast Asia. And there were two different reasons for that. One is that our sequencing partners have sort of an interest in that region. And the second one was because of how prior projects had performed in covering the diversity of that region. So what you see here is a study which came out in 2014, which essentially looked at the 1,000 Genomes Project. So the 1,000 Genomes Project sequenced about 2,500 samples from across the globe. And so far, that is the largest study which did whole genome sequencing for a large number of populations. What this particular report did was analyze how the 1,000 Genomes Project compared to something called the Human Genome Diversity Panel and the Pan-Asian SNP panel. So
I'm sorry, Akrosh. I think your slides crashed because we still see GA100K goals slide. Oh, yeah.
Okay. All right, let me try this again. Do you see the 1,000 genomes versus 8 GDP?
Yes, okay. We can see it now.
Thank you. Okay, so as I was saying, the Human Genome Diversity Panel and the Pan-Asian SNP panel have been two large studies that have tried to encompass the diversity across the globe, but they were done on microarrays. So we have only genotypes for a limited number of markers. And what this report tried to do was to assess how does the 1,000 Genomes Project do compared to these earlier studies. So in panel A here, what you see is green dots and marks are essentially from 8GDP or PA-SNP, and the red marks are for samples that were included in the 1,000 Genomes Project. And you can see a few things immediately, right? So this region here which is Oceania, those samples are not included in the 1,000 Genomes Project. You can also see there are parts like this region here that is not included or covered by the 1,000 Genomes Project, right? So this is just a principal component analysis. So what you see here on the x-axis is principal component 1, and on the y-axis is the principal component 2. Principal component analysis tries to find a direction or an eigenvector that shows the maximum variance in your data set, right? Okay, so if we remove some of the populations that are well covered and you look at panel B, and if you focus, we have limited ourselves to East Asian, Southeast Asian, and Oceanian populations, you will see that the 1,000 Genomes Project did not cover Oceanian populations, and it did not do a good job covering Southeast Asian populations. And this becomes more apparent or most apparent in panel D, where we are only looking at Southeast Asian populations. And what you immediately realize is that the 1,000 Genomes Project actually covers a very small portion of the genetic variation of these populations. So we actually made a concentrated effort to sequence a larger number of samples from South Asia and Southeast Asia for this pilot genome project. All right, so as I said, we had 1,739 samples from 219 populations. 1,667 of these samples were unrelated. we required at least a coverage of 20-fold from these samples. And in fact, most of the samples were covered to more than 30x. We included 119 samples that were sequenced in the 1,000 Genomes project at a much lower coverage. So somewhere between 4 to 7x was the coverage in the 1,000 Genomes project. And we included it primarily for quality control. We used the GATK toolkit to essentially do joint calling, which means we were interested and we called the genotype for every sample at every variant location. And in the end, that call set consisted of about 63 million single nucleotide variants and about 4 million indels. And when we looked at the concordance with the 1,000 genomes genotype in these 119 samples, the variant concordance was close to 99.63%. Now, what's interesting is, as I mentioned in the 1,000 Genomes project, these sequences of these samples were sequenced to between 4 to 7x. And what we see immediately is the places where we disagreed with the 1,000 Genomes project were places which were essentially singletons. So in these 119 samples, the most rare events, right? So which were only seen in one sample, that's where we disagreed, which is not surprising, right? At 4 to 7x of coverage, the power to call heterozygous variants is extremely low. We also included the genome in a bottle samples as part of our call set. And for those of you who do not know, Genome in a Bottle is another consortium. And there, instead of including a lot of samples, they are trying to take a few samples and create the best call sets possible. So they use not only short read sequencing, they will use all different flavors of sequencing. They will run multiple tools and try to identify a consensus call set. And when we compared our results in those samples, we had a precision greater than 99.99% and a sensitivity of close to 91%. So, and that sensitivity basically shows you the limits of short read sequencing. And in case I wasn't clear, we used Illumina sequencing to sequence these 1,739 samples. All right, so this is an example of a simple variant metric. So what you have here are the individuals collated in these 7 superpopulations, right? So these are somewhat, you can refer to them as continental populations. And what you have on the y-axis are the number of substitutions per genome. And you will see immediately that these can range between 3 million for European samples to up to 4.3, 4.2 million for individuals of African origin. What you also see immediately is...
I'm sorry to interrupt again. I think the slides are much delayed due to you sharing the webcam. Maybe we could try, because we cannot see your current slide. Maybe
Okay.
you can try freeing this. Yeah, so closing the web. Yeah, just can you close it so that you are not sharing it anymore?
All right.
All right.
Is it closed?
Yes, now I think so.
And now you can see my slides.
Yes.
All right, thank you. Okay, so what was I saying? Okay, yeah, right. So what you also can see here very clearly is that there are a lot of outliers, right? So you can see this smear of our outliers, outlier samples when you look at South Asia or Southeast Asia, and that is reflective of our sample bias, right? So we have a lot more samples, but it is also reflective of the extent of diversity in several of those populations, and I'll talk more about that. This is another simple metric that you can think of. These are the mutations that are found in a single individual, right? And typically for all of us, about 1% of our substitutions or mutations compared to the reference genome are ultra rare. They can be singletons, but at least they are ultra rare. And even though all of us have only 1% of mutations that are ultra rare, when you look at the call set as a whole, there is a huge abundance of rare variants. So what you see here on this slide is on the x-axis, you see how many times do I see this alternate allele. And on the y-axis, what you see is the number of substitutions detected in our call set. And you will immediately see that the largest number of variants that we detect are the ones that are ultra rare or singletons. So they are only seen in one sample in our data set. And that is where we find most of the novel mutations when we start comparing to existing databases, right? So here we are comparing our call set to the call set from dbSNP, from 1000 Genomes Project, from Exome Sequencing Project, and GNOME-80. And as you can see, 25% of our alleles are novel and when they are singletons. And this number decreases as we go further. Now, this tells you on one hand, it gives you some idea of the value of our call set. On the other hand, it also tells you that the 1000 Genomes Project did a pretty decent job of covering the allelic diversity in a majority of the populations. But as we all know, most of the variation among humans is within populations and not between populations, right? We are extremely similar to each other. But if we want to focus on the differences, we can use analyses like principal component analysis. So that's what you see on this slide. Again, the x-axis is PC1 and y-axis is PC2. And what you clearly see in our data set is that the African individual, PC1 basically separates the African individuals from the rest of the genomes, and y-axis you can see a smear of ancestry, of Asian ancestry, as well as oceanic ancestry. And that is consistent with the idea that most or modern humans originated out of Africa. And we see similar results if we start looking at what's called the fixation index. So fixation index is essentially looking at pairwise differences between all samples and then calculating the ratio of the differences between versus within populations. And again, what you see here, the African individuals are outliers, again, supporting the hypothesis that most of the allelic diversity among modern humans resides in Africa, and all of us essentially started out from that region. We can also, because our data set was very rich in certain parts of the globe, we could focus and look at this fixation index among several populations. So when we focus at, let's say, the South Asian groups, you will see the groups from Pakistan share close affinity, suggesting recent shared history. And then when you look at Indian populations, two populations stand out. And those are the Jarawa and the Onge populations. These are tribal populations, indigenous populations from the Andaman Islands. They seem to be very similar to each other, but they are very different from all other South Asian ethnic groups. This reflects both a long period of isolation in the Andaman Islands, as well as increased genetic drift due to the small population size. We can also look at other parts of the globe. For example, let's look at Northeast Asia. And you will immediately see the populations there have low FST values. And this suggests, again, recent shared history. When you look at Southeast Asia, there are some surprises or interesting things to look at. The populations that have higher or outlier FST values are actually what's called anthropologically termed as Negrito populations. These are individuals typically who have darker skin. And what you see here are two different populations, Eta and Ati. They are from Indonesia, and there is this population from Malaysia, and they stand out in terms of FST values. Interestingly, if you compare these populations to their geographical neighbors, they share a lot more similarity with their geographical neighbors than with each other, supporting the hypothesis that dark skin is essentially an environmental adaptation as opposed to reflective of shared ancestry. Fixation index is just one way of talking about population stratification, right? Another interesting way is to characterize it by the degree to which genomic segments are inherited. So instead of looking at individual SNPs, you can ask, are there long haplotypes that seem to be shared among individuals belonging to a specific population? And in fact, if you think about genome-wide association studies, the goal there is to identify if the cases or one group has a haplotype that's shared compared to the other group. So what you see here are the populations on the x-axis, and on y-axis, what we call an IBD score, so identity by descent score. Just so that we can be on the same page, such scores have been calculated in the past in several other studies. And the typical scores for Finnish and British populations are about 1.465 for Finnish and 0.817 for British groups. Now, Finnish populations have come around from a very small founder population, right? And they are frequently used for genome-wide association analyses. And the reason for that is if you come from a small founder population, you are bound to have long tracts of genomic segments that are shared between individuals, and that increases the power of a genome-wide association study. So you can actually create or identify associations with a much smaller sample size. So as you would expect, as I was talking about some tribal populations that have been isolated, right? So specifically Onge and Jarava, they have huge, really high IBD scores. But what's interesting is a large number of Asian populations from large metropolitan cities have IBD scores similar or greater than Finnish populations. And that is an interesting opportunity, right? Because we have a unique opportunity now to study varying phenotypes. There is a preponderance of rare diseases because they have large census populations, and they have high enough IBD scores that these populations, there is a huge opportunity to actually do genome-wide association studies and for many complex traits. The other analysis...
I think you're frozen again.
All right, let me try. Is it better now?
Yes.
Okay. Thank you. So the other analysis that we looked at was, as I mentioned earlier, about human knockouts. So we did look at homozygous loss of function alleles. So homozygous loss of function alleles found in humans provide us an opportunity to assess the phenotypic effect of specific gene losses. And they can provide, in certain cases, important information about opportunities for therapeutic intervention. So what you see here is basically the proportion of genes with at least one high confidence protein truncating variant. And if you further focus on it, you will immediately see that most of those protein truncating variants are heterozygous. So it's not a complete loss of gene function, but we did identify a small subset of homozygous protein truncating variants. Most of them have been reported in other studies, but we identified about 121 high confidence protein truncating variants that were novel. And just to give you an example, this particular variant was seen in the gene ABCA7. And there have been other loss of function mutations shown in gray here, which have been reported by other studies, but we found a novel protein truncating variant in the C-terminal ABC domain. Interestingly, heterozygosity for loss of function alleles of ABCA7 has been shown to increase susceptibility to Alzheimer's disease in European populations. So this has not been replicated in any other population as far as I know. But this is just an example of the sort of interesting things you could take from this data set. So the primary reason we wanted to do this project and the primary reason we came up with this call set was because we think of this as a resource, right? So now I'm going to give you a few examples of how you can use this as a resource and primarily a clinical resource. So the first example I'll give is when we looked at a data set of 152 exomes from patients who had been diagnosed with MODY. MODY is the name given to a collection of inherited form of diabetes, typically monogenic, that usually develops in adolescence or early childhood. It stands for maturity onset diabetes of the young. And the name was given in the past because it acts like adult type of diabetes. So it acts like type 2 diabetes, but it is found in the young. It limits the body's ability to produce insulin. When our bodies don't produce enough insulin, it increases your blood glucose levels. High blood glucose levels lead to diabetes, right? So we looked at a cohort of 152 exomes, and the question is, are there damaging mutations that would tell us something about, or either we can say with confidence that these patients, or we understand the molecular etiology of the disease. And one of the things that you want to do when you do something like this is immediately identify variants and then remove the variants that are common in so-called healthy populations, right? And typically we would use a large database to do that. So here you see when we used GNOME-AD to remove what's common variants in healthy populations, we were left with, on an average, 914 mutations that we had to then further dig into. But once you include the Genome Asia variant call set, and because these samples are from South Asia, the number of mutations we then had to dig into reduced by 400. So we had only to look at 533 variants for further analysis. So that's one example of how you can use the Genome Asia call set to prioritize mutations in a disease, and specifically in a rare disease. Let's look at another example. What we did was we took our call set and we annotated it. We annotated the mutations on whether they were present in clinical databases, specifically two of them: Human Gene Mutation Database, which identifies disease-causing pathological mutations, and ClinVar database, which identifies pathogenic variants. This analysis identified about 732 variants in 514 genes. We found that 38 of them had an allele frequency that was higher than 0.15% in our call set. So we then sat down and reviewed these 38 variants against the criteria defined by the American College of Medical Genetics. And we ended up reclassifying 11 out of the 38 variants as not being pathogenic, right? So you can see how basically having variant call sets from diverse populations now allows you to go back and revisit your knowledge base from, let's say, people of European descent and reclassify those variants. This is another example of how this data set can be used. And this goes back to pharmacogenomics, right? We all know that there are specific SNPs that have been associated with adverse drug response. For example, let's take warfarin. Warfarin is an anticoagulant. And what you can see here immediately is that the drug response, the adverse drug response, can be different among different populations. And so the dosage of warfarin has to be administered and dependent upon the genetic makeup of the individual. And you can clearly see there is a broad spectrum of that drug response, adverse drug response in these populations. All right, so I mentioned that this is a resource. So currently, there are a few ways you can interact with this resource. So you can go to this browser link, and in the title bar here, you can specify your favorite gene or your specific region. And for most genes and regions, we will then present you with a table, which will have the mutations that were identified in Genome Asia, as well as some quality metrics or annotations about those variants. The other way you can interact with this call.
We cannot see the next slide. Yep.
All right, sorry, hold on.
All right, now we can see the browser.
Okay, I'm not really sure why this is happening, but okay. Can you see my slide?
Yes.
Okay, so I talked about the browser, but basically you can go to this link and enter your favorite gene or your favorite genomic region. And within reasonable, like there are certain regions we do not show currently, but for most regions, you will then be able to see a list of the variants in Genome Asia, as well as some annotations and metrics associated with them. The other way you can interact with this data set right now is if you are doing imputation. So let's say you have a genotyping data set of individuals of Asian descent. You can then go to the Michigan imputation server, and as a reference panel, you can use the Genome Asia pilot call set. So that will allow you to basically identify or probabilistically call the SNPs at locations that are not covered by your panel. So as a last example of the utility of...
Excuse me. I'm so sorry for these technical issues. I think now it fully crashed because we cannot see anything.
All right, hold on. Can you see my desktop now?
Now we can.
Okay, and now you can see my
Yes.
slide. Okay. I'm sorry about all these technical troubles. All right, so the last part that I would like to talk about has to do with some of our analyses looking at archaic human admixture. And this work was led by Jeff Wall from University of California, who is an expert in this region. So before we started on this study, we sort of knew a couple of things. We knew that non-African individuals inherit about 2% of their DNA from Neanderthals. And Denisovans are another group of hominids that contribute about 4% to 5% of the DNA in Melanesians and Aboriginal Australian individuals. And there have been several studies that have shown that the introgressed Neanderthal regions are generally deleterious in modern humans, and they've been associated with a few health conditions. And if you look at functional parts of the genome, you see that they are more rare, so they are under negative selection. So we took our call set and we asked the question, what is the fraction of the genome that is probably contributed to by the Neanderthal admixture, can be attributed to the Neanderthal admixture? And we found that the highest amounts of Neanderthal admixture were in Papuan and Melanesian genomes, so somewhere around here, followed by East and Southeast Asian genomes, South Asian genomes, European genomes, and Middle Eastern genomes. And that confirms the current knowledge base, right? So we found similar numbers as what you would expect based on prior studies. But when we started looking at Denisovan admixture, we again found that the Denisovan ancestry was highest in Melanesians, but we also found another population, which is the Eta population from Indonesia, which had similar amount of Denisovan ancestry as the Melanesians. We found it in intermediate in Ati, which is an Austronesian group, again from Indonesia, and Indonesian islands of Flores. So and it was lower in most other parts of Southeast and Southeast Asian populations, though it was significantly greater than 0. So the high levels of Denisovan ancestry in Melanesians and the Eta are consistent with an admixture event into a population that is ancestral to both, right? However, there are two different lines of evidence to suggest that the ancestors of Eta experienced two different admixture events. One of them is that if you look at Denisovan haplotypes that are unique to Eta, they tend to be significantly longer than the putative Denisovan haplotypes shared between Eta and Papuans. The second is that Eta are, as I mentioned before, more similar to their neighboring or geographically neighboring populations, right? So which means that there has been recent gene flow into the Eta from non-Denisovan populations, or populations that have very little Denisovan admixture, which in theory should decrease the amount of Denisovan admixture. However, as I mentioned, the amount of Denisovan ancestry or estimate of Denisovan ancestry in Eta is very comparable to those among Melanesians. So we have sequenced, a few samples, a few Neanderthal samples. I mean, other groups have sequenced a few Neanderthal samples and Denisovan samples, so we can actually calculate or make estimates about Neanderthal ancestry and Denisovan ancestry. But what about unknown archaic or hominids that have not been discovered yet? So the way you try to look at data and try to infer these admixture events is by trying to identify long diverged haplotypes, right? If all members of a population and there is a single outlier haplotype, then that stands out, and that is typically called a ghost haplotype. And we tested a pipeline which was able to predict the estimates for Neanderthal and Denisovan ancestry really well. And then we asked, okay, are there other ghost haplotypes in other populations around the world? And we found evidence for at least one archaic admixture event in sub-Saharan Africa, with the strongest signal in what's called the Khoisan, which is a hunter-gatherer population in South Africa, and the Pygmy populations in southern and central Africa. The locations of these putative admixture tracts can be weighted against functional regions of the genome, and we found, similar to the analysis done for the Neanderthal introgression, we found that these regions are under mild deleterious or negative selection. All right, so what's the road ahead? So there are a few different things that we are working on. One is most of the analysis I talked about is focused on single nucleotide variants, and we are now working on a call set for structural variants. We are focused on breakpoint resolved set of variants, where we intend to compare it to known databases. There are now 2 or 3 big databases of structural variants that we can compare to. We'll focus on loss of function, gain of function, or duplication of large genes. And we plan to comment upon some repair mechanisms that would go into into basically the formation of these structural variants. I also talked about some of the biases in the reference genomes itself, and we are now working on, actually my group is leading the efforts to develop reference genomes from Asians. And the way we are doing this is by a combination of long read and short read sequencing. And of course, we keep on doing more and more sequencing. So as of now, I think we have sequenced more than 10,000 genomes, and the next analysis of close to 3,000 whole genomes, where the focus has moved more towards the North and Northeast Asia. Can you hear me?
Yes, we can hear you, and we can see the road ahead.
Okay. Yeah, there is some feedback. Anyway, so yeah, the focus has now moved a lot more towards North Asia and Northeast Asia. So the next call set will be focused a lot towards those populations. And, if you're interested in working on these kind of data, we are always looking to hire. So please get in touch, and thank you. And I'll take any questions that you have.
Thanks a lot for this great talk. I'm very sorry that we had so many technical issues. Yep. All right. So we have some questions. First would be from YouTube. How the results of telomere to telomere consortia, so long-read assembly of human genome, could affect the results of the current project?
Right. So the telomere to telomere project, it is a very interesting project, but in terms of the genetic variation, unless we want to look at variation in the telomeric region, the limits are due to short read sequencing as well, right? So even if we had a reference genome where we had telomeric to telomeric variation, unless we are willing to investigate long read sequencing for our samples, it would be difficult to interrogate variation in those regions. However, we do plan to employ certain techniques that they have developed when we look at constructing reference genomes.
All right. I think we lost the slide on identity by descent. But there was a question, what is the reason behind the high identity by descent score for the Asian metropolises?
So can you see the slide?
Yes, we can see it now. So okay. What do you think is the reason?
Oh, so there are several reasons, right? The biggest reason is they have small founder populations. So human history is a story of migrations, right? So we all start from Africa and small groups of people leave and move on, right? And do their own thing. And a lot of the rationale is that we move on. We have very small founder populations. And after the advent of agriculture, there has been a huge explosion in population. So if you had and that's probably it, right? And in certain cultures, consanguinity might play a role, but I would say small founder populations is the biggest reason.
Mm-hmm. So there's one more question. Thank you for a great talk. It's a great resource. My question is about the cancers. There are some malignancies that are more prevalent in Asian populations. Have you looked whether some of the frequent mutation in those are present in your data set?
Right. So that's a great question. And no, we have not. And primarily because the first round of samples that we were collecting, we did not focus on any disease per se, right? So we were looking at somewhat healthier populations, you would say. But no, that is on the list of things we want to do. We did identify, so we did look at genes that have already been shown to increase risk for cancer. And we did identify novel mutations only found in the Asian populations. Like, for example, in BRCA1, there was a truncating mutation that had not been seen so far. So, for example, that. So we did look at the risk providing or risk genes and find some novel mutations, but we have not done a deep dive on that. But that's a great question, yeah.
Okay, I think we have time for one more. So what are the challenges with working with such a high number of different populations? You mentioned 219, especially when building a new reference genomes.
Right. So one of the challenges is which populations do you select for building a reference genome, right? I mean, there is limited amount of money, there is limited amount of resources, there's limited number of people we have to work on this project. So the question is, which populations do you select to build a reference genome from? And one way to think about it is to find populations that are most divergent and sort of use them, right? Because you want something that's not like the reference genome, and you want to build a reference genome from that. The other way to think about is which is the reference genome I can build, which will help or which will be more useful for the most number of people, right? So if there is a population that has a very large census size and I build a reference genome for that population, they are more likely to get medical benefits out of that. So there are lots of different ways to think about it. And so far, we haven't really come up with a single answer. So we are exploring all sorts of answers. But my guess is we will end up building 9 or 10 different reference genomes. As far as technical challenges, there are challenges with reference bias. There are other challenges which I'm not the right person to talk about, which are related to trying to get samples and coordinating IRBs. And, there are a lot of political and other challenges when you start looking at so many different populations. But I'm not the right person to speak to that. But I believe those challenges are significant.
Right. It is very hard to even think about looking at such wide, not only geographical area, but a high number of different populations. And I suppose there was also a question which is connected to that, about encountering obstacles when inviting participants to this project. In general, is there something we as researchers can do to help increase participations in projects like this?
Well, yeah, if somebody approaches you and asks you if you are interested and if you have samples or analyses to contribute, you can always say yes. But in general, we found it difficult, as you mentioned, because a lot of countries have their own genomic projects, right? So UK has its own project. I believe Qatar has its own project. Singapore has its own project. So it's really challenging. And each country has its own political landscape and laws related to how samples should be used, how the research has to be provided back or not. And it is challenging, but that's where I appreciate there are a lot of other people beside me who are willing to look and work on those challenges, right? So I can only speak to the technical challenges, and those are, I would say, not as bad.
All right. Yeah. Thanks a lot. That was an amazing talk, and it covered a wide area, and I think it is very important for us to hear about projects like this. So thank you, even though we had some technical difficulties.
Yeah, I apologize for the technical problems, but it was great. Thank you so much. Thank you.
So the next talk we have prepared is a talk, I hope I'm pronouncing correctly, of Lucia Ximena Alva Caballero from National University of Trujillo in Peru. And it's going to be a talk about, we are staying with the genomic diversity topic, so it's going to be a talk about using SNP data to investigate chromosomal races of house mice. Lucia, the floor is yours.
Okay, thank you. Good afternoon, everyone. So I'm Lucia Alba. I'm from Universidad Nacional de Trujillo, and I did this work along with Jonathan Hughes and Jeremy Searle from the Searle Lab in Cornell. Sorry, can you all hear me?
Yes, we can hear you and we can see your slides.
Okay, great. And the talk is titled Using SNP data to investigate chromosomal races of house mice. So, on the last few years, the DNA sequence cost per genome has been dropping down, and that, along with new techniques for genomic analysis, has given us the potential to answer fundamental evolutionary questions that we haven't been able to answer before. One of these questions is, how do new species originate? So on Earth, we have this huge biodiversity. However, we don't know exactly how it was formed. We have many hypotheses, and one of them is through the evolution of karyotype. Here we have the karyotype of humans and the karyotype of chimpanzees, and we can see that one single difference on its karyotype gets at two completely different organisms at the end. However, today we're going to see the case of the house mouse. There are high rates of chromosomal evolution in the house mouse. Here we see the karyotype of the standard race, which is conformed of 40 telocentric chromosomes. However, here we see the karyotype of an specimen of Italy, and it's completely different from the one of the standard race. Here we can also see the karyotypes of two individuals from the island of Madeira, and both of these individuals have completely different karyotypes. However, all of these specimens have the same morphological features and behave the same. So having different number of chromosomes makes for organisms harder to interbreed. And when we see this diversity of karyotypes in one species, we ask ourselves if we are seeing the first stages of speciation in the house mouse. So there are different processes that lead to chromosomal diversification in the house mouse. One of them is the Robertsonian fusions, in which the chromosomes break at the centromeres and the long arms fuse to form a single large chromosomes. We also have warts, where are whole arm reciprocal translocation. As you can see here, one of the chromosomes break and becomes a single telomeric chromosome, and the other one fuse at the one that was previously broke to form one single large chromosome. And here we can see a meiotic pairing configuration of an heterozygous individual. Here we can see that we have one single telomeric chromosome, one large chromosome comprised of two chromosome fuses by the centromere, another large chromosome, and another single chromosome. And we see this happen all the time on the chromosomal races of house mice. So where are these chromosomal races located? They are located all across Europe, from the countries at the north of Europe to one country in the north of Africa. However, today we are going to see the chromosomal races that are on the island of Madeira. Why are we choosing Madeira? Because the timing of colonization is well known. We know that the first mice that came to the island came along with the Vikings, and then they came with the Portuguese colonizers. In Madeira, we also have 6 races with a high degree of karyotypic variation. These are all the races. And also, Madeira system is clearly defined geographically because it's an island. And the question that we asked ourselves in this research was how has karyotype change affected the different chromosomal races of mice in the island of Madeira? And the goal was to investigate the relationships between the races using SNP data. So we wanted to know if these races are still able to interbreed with each other, even though they have karyotypes that are very different from each other, or if they have reproductively isolated. So the data collection was done on the island. Each of these dots are the different locations in which a specimen was collected. Then those specimens were brought to the molecular lab to extract DNA from their tail tips, from their slipins and their toe clippings. Then we use SNP data to obtain a PCA and phylogenetic trees. So SNPs are single nucleotide polymorphisms that occur across the whole genome, and we use a high-density mouse universal genotyping array called MegaMuga, which provided us with around 74,000 SNPs. So we extracted the SNP data using a Blink. We had data from different races of chromosomal mice from all across Europe, and we extracted the data from the ones of Madeira. Then we converted that data to a FASTA format, and we also obtained a PCA using 2 programs, which are SNP Relate and SNP Philo. Then we got an alignment with only the variable size using IQ3, which is a model, in this case was GTR plus ASC, which takes into account the assortment bias, which is very important for this project because we are working with the SNPs. Then we ran an statistical analysis, again using IQ3, so we could get a consensus tree at the end. So one part that is very important in this project is that there is a difference between telocentric and centromeric data, because we are working with a karyotype in which chromosomes have fused at the centromere. and also because of recombination, in which chromosomes exchange genetic information. So, however, in the centromeric area, there is a suppressed recombination. So because of that, we wanted to get data from the centromeric area. So we extracted SNP data using Blink. We chose the mice from Madeira, and when we were extracting the data from the centromere, we noticed that Megamoga has given us a higher density of SNPs in the telomeric area. So we couldn't get too close to the centromere, or otherwise we would lose a lot of information. So we tried to get information that was close enough to the centromere, but it was enough to get an accurate phylogeny at the end. So this is the first result that shows us that P-pod seems to be distinct among the other chromosomal races. Well, as we can see in the PCA, P-pod is very far away from the other races. Here we can see the race of PSAN and PSBI. Even though they are clustered together, there is no overlap between them. However, if we see at the races from the west of the island, we see that they are clustered together and there is a lot of overlap between them. So that suggests us that the races from the west of the island are still interbreeding with each other. Now we're going to talk about the telomeric and centromeric SNPs that generate different phylogenies. Here we have the centromeric tree, in which P-pod and PSBI form a paraphyletic group, and this makes sense with the PCA that we obtained before. However, if we look at the telomeric tree, we can see that P-pod and PSBI are together, and that doesn't make sense if we see the PCA that we obtained before. So that can tell us that it's probably the centromeric SNPs has provided us with a more accurate phylogeny than the telomeric ones. However, both of these trees show us that there is still some gene flow between PADC, PADC, and PLDB. And why is that? So the program that we use, IQ3, doesn't account for hybridization between individuals. So that's why we got a messier phylogeny at the end between these races. However, in the telomeric tree, there are more groups, and the chromosomal races hadn't clustered together, and we have a more neater, a more cleaner phylogeny on the centromeric tree. So we are going to take this data in future research. And the take-home message for this presentation will be that in the PCA and the centromeric tree, we got P-pod and PSBI as paraphyletic groups, and the trees show gene flow between the western races of the island. Now, we have a lot of hypotheses of why this happens. So it's important to take into account that this is the island that we're working on, and in this island, there are mountains that go through the center of the island, and also there is some elevation between the cities. So we think that the P-pod race that it's located on this part of the island has reproductively isolated from all the other races because there is some geographical barrier that happens between the towns. There is also important to notice that house mice don't have a great distribution range. They mainly live in the house where they are found. They don't go too far from there. So there is also an important thing that might have contributed to the isolation of some of the races at the north part of the island. As we can see here on the west part of the island, there is almost no geographical barriers, and also there is a lot of a heavy flow in between the towns of this part of the island, so that might also contributed to the still gene flow between the races that live here. Another hypothesis is that is the origin of the mice. We know that some mice have come with the Vikings and have stayed in the island for a longer time than other mice that came along with the Portuguese. So that might also have contributed to the isolation of some races and the still gene flow between other races. For the future, we want to use the SNP data with a bioinformatics program that can take hybridization into account. We also want to redo the fieldwork to see if anything has changed between the years when the fieldwork was first done and now, because the house mice have a higher evolution of a karyotype, so we wanted to know if anything has changed. And I would like to offer my special thanks to the SEER Lab at the Department of Ecology and Evolutionary Biology at Cornell, to CONCITEC for giving me funds, to the principal investigator, Jeremy Searle, to the PhD student, Jonathan Hughes, to my mentor, David Chang, and to the REPO program. So,
thanks a lot for the talk. We have some questions. All right, let's start maybe from very general questions. So you mentioned that the races of mice migrated from Europe. Are the populations right now on Madeira still genetically similar to European equivalents of the same races? In other words, can we extrapolate the findings to mice from the European continent?
Okay, yeah, the mice that were found on the island of Madeira have emigrated from Europe. However, there wasn't any chromosomal race that got to the island. All the mice that emigrated first were from the standard race, and it started to develop the karyotypic changes when they were already on the island. So all of these races, even though we can track back from where they are, we have seen that there are some races that are more genetically similar to races found in Denmark, and there are also some races that are more genetically similar to some mice found in Portugal. So even though we can look at that, all the karyotypic changes happened already on the island. All the mice that came to the island were from the standard race.
All right. So due to the island geographical conditions, there's an isolation that is promoted between the center and east mice. Can you tell us more about this? Because between west and east mice, I think it's pretty obvious, but between center and east?
Okay, yeah. So between the center and the east. One thing that is pretty important is that house mice move with the people, so move with humans. So what happens is that if there is, for example, a heavy trade flow between humans in some part of the island, we are gonna find more genetic flow between the mice of that part of the island, which is what happens on the west part. Here, there are not that many human populations, there are not that many towns, so that's why we haven't been able to found many chromosomal races or even many individuals in this part of the island. Funchal, which is the main city of the island of Madeira, for example, only have the standard race, and we think that that happens because of the heavy trade flow between Funchal and Portugal. So that's one reason. Also, here there are some towns on the northern part. However, those towns seems to not have enough trade flow with all the other towns as in the west part of the island. So one thing that I want to make clear with this is that the house mouse moves with the human population. So if we found humans in this part of the island, it's probably that we are going to find mice on this part of the island. And if the humans in one part of the island move a lot and trade a lot with other populations on the island, then we're going to find a more flow between the mice on the that part of the island.
All right, thanks a lot. So we have one more question. Very nice work. Is the number of SNPs used to build the two trees very different? And could the difference be causing the differences into phylogenesis? If there is a significant difference, have you tried to test this, for example, by downsampling the number of SNPs?
Yeah, there is a difference, mostly because Megamuga has almost the double of SNPs in the Telomericc area than in the Centromeric area. So we, for example, if we want to get, I don't remember quite exactly the number of SNPs, but let's say if I wanted to get 5,000 SNPs from the telomeric area, I will have to get like a 10% 10% SNPs from that area, and I don't know, 30% of SNPs from the centromeric area to get the same number of SNPs. However, what we did just to make sure that we weren't like these results weren't the result of just the different density of SNPs in this area was that we did the PCA also with only the telomeric data, and we can see that here. I think I have, okay, here. And the SNPs from the telomeric area in a PCA also supports the theory that the pivot race have become isolated from all the other races. And if we compare this PCA, which was from the telomeric SNPs, to the PCA that we got from the centromeric SNPs, we can see that they are quite similar. So the only problem that we got with the telomeric data was while doing a phylogenetic tree. As I said before, the reason that we choose to work with centromeric data was also because of the recombination that happens a lot in this area, and that will allow us to get like a messier phylogeny at the end. Mostly because we are trying to look at this area in which the chromosomes have fused.
All right, thanks a lot. It is very great to see a study, especially that it's a very good example of migration. And we are very happy that you decided to submit this abstract and give this presentation. I think we're going to move. There's no more questions. If there are some more questions, I invite you all to comment on YouTube and on Discord. So we're going to move to the next speaker. Let's see. Yeah. Okay. Everything should be ready. So the next speaker, I'm sorry if I'm mispronouncing. Is Ragir Salkin. Am I close? Maybe no. And he's going from Khulna University of Bangladesh. And he's going to tell us more about the dietary fish lipids for protection in silico screening. The floor is yours. I'm not sure if we can hear you though.
Can someone confirm if they can hear me?
Okay, now we can hear you.
Okay. I presume you can see my screen as well?
Yes.
Okay. Well, should I start?
Yes, please.
Okay. So good afternoon, everyone. I'm Ragir Salkin from Khulna University, Bangladesh. I'm an undergraduate student of biotechnology, and today I'll be presenting on the heteroprotective and anti-inflammatory activities of bioactive dietary fish lipids, which I have conducted an screening study on. Here are my co-investigators and the presentation contents. So if you look at the background, so atherosclerosis refers to a complex vasoconstrictory condition, which encircles a lot of pathophysiologies, including oxidation, inflammation, and homeostatic dysregulation. And it's highly linked to cardiovascular and pulmonary health, which is rather important, especially amidst the COVID situation we're going through right now. So what happens in case of atherosclerosis is that when our immune system is hyperactivated, or the pro-inflammatory pathways are overexpressed, they tend to accumulate in the blood circulatory system. And as we know, there are also a lot of lipid floating around in the circulatory system as well. So the lipid and the inflammatory cells together form plaques or lesions in the blood vessels, which ultimately constrict the blood flow and result in cardiovascular and arterial complications. So if we look at the overall procedure, we'll also see the current therapeutic approaches that are being taken for atherosclerosis and atheroprotection. So the current available commercial drugs usually target 4 different pathways, 4 different key regulatory proteins that are the angiotensin converting enzyme 1, L-type calcium channel, LTCC, cyclooxygenase 2, and thromboxane synthase. So cyclooxygenase 2 is more involved with the arachidonic acid metabolic network, which is more involved with oxidation, inflammation, and cancer. On the other hand, thromboxane synthase is rudimentary to the blood coagulation cascade and blood clotting. So as you can see, these proteins are not independently acting. They're actually interconnected by feedback loops and interconnections and secondary metabolic communications. So the problem arises right there is that conventional therapeutics usually target one protein or one pathway, and they neglect this inter-channel or inter-network communication, which results in side effects and crosstalks that are not considered when designing the drugs. So key objective of this study is to find probable drug-like compounds that can actually work on multiple pathways and multiple networks of atherosclerosis and atherosclerosis, which ultimately help in cardiovascular health and overall coronary complications. So fish fatty acids, or you may know them as fish oil supplements, have been touted for their cardioprotective and inflammatory roles for quite some time now. But the problem is that while there are some preclinical studies, the results or data is substantially less to be significant in case of drug designing, drug discovery. So especially if we consider these pathways and proteins that we've mentioned, there are not any significant data on how these fish oil supplements interact with these proteins. So based on these information, the core objective of our study was to provide a molecular insight into how the atherosclerotic activities of these bioactive fish acids can be translated when we consider multiple metabolic pathways. And secondly, we want to go the drug discovery route and see if any of these or which of these atherosclerotic compounds from fish oils can actually act as specific inhibitors of the 4 key regulatory proteins in the atherosclerosis pathway. So our experimental section comprises of 2 different phases. The first one was a rudimentary systematic review, where we found 32 reported fish oil supplements or fish oil compounds that are reported for their cardiovascular activities. And in the second section, we took these reported compounds, some reported drug molecules for positive control, and to identify any false positive, we also took some decoy molecules, and of course the receptors or the protein structures that we wanted to study. And after undergoing some preparations, we went for a 2-phase molecular docking simulation, and after that we went for molecular dynamics and molecular mechanics finally followed by metabolic network simulation and statistical analysis. So we'll jump right to the results section. So the first objective was to see if these compounds we're studying, if they're safe or not. There's a lot of data on the screen, but what's important to see is that most of these compounds have very high gastrointestinal absorption, which is very important for drug-like compounds, and they're also very good blood-brain barrier penetrance, which is very good. And secondly, we went for the bioavailability score, which is optimal is 0.55, but in this case, we can see that most of the compounds have a very high bioavailability of 0.85, which means that they are very good drug-like compounds, actually. And secondly, we look at the in silico admin properties, especially toxicity profiles of these compounds, particularly the toxicity, toxicity, carcinogenicity, and allergenicity. So you can see some of the compounds do, they have been predicted to be allergenic, but we've incorporated them as they've been used as dietary dietary supplements for some time. But we did take the carcinogenicity index very seriously, and the three compounds that were predicted to be as such were excluded from further studies. Here is a more summarized view of the in silico admin studies, and we can see this drug-like compounds here, which are the eicosanoic acid, eicosanoic acid, nervonic acid, and DHA and DPA. The diagram represents the egg yolk, means that these are very good blood-brain barrier permeants, whereas the white part means that they're very good gastrointestinal absorbents. So after we've identified which compounds are good drug-like compounds, we went for the docking simulation. Here you can see in case of the L-type captain channel protein, we identified the lipid binding cavity and docked our 29 compounds into the cavity and identified the top 5 compounds, as well as compared them with a positive drug that in this case was a diltiazem, which is an antihypertensive drug. Here you can see that the best compound, trexamide acid, was a very good, was far superior than the available drug in our simulations. One thing is to be observed here is that traditional or conventional idea is that looking at any of these bioenergetics involved mostly contributed by the hydrogen bonding. But in this case, since they're mostly fatty acids in structure, the drug or rather the lipid protein interactions are most dominated by hydrophobic interactions in these amino acids. Even then, we can see that they cannot perform the conventional drugs, which are mostly ionic or hydrogen bonding in nature. Similarly, in case of angiotensin converting enzyme, we can see the top 5 converting performing compounds, but in this case, they fail to outperform the best drug, which is ranitidine. And the yellow one is the angiotensin 1, which is the native substrate. So these compounds need to substitute this native substrate in the binding cavity to exert the therapeutic effect. So they are capable of doing that, but they're not outperformers of the conventional drugs. However, the scenario is drastically changed when we consider cycloxanate 2, where the top 5 compounds all actually far outperform the conventional drugs. Similarly, in case of chocoboxin A synthase. So the docking simulation actually gives us a probable idea of the how should I say, the complete model of the enzyme and ligand fitting, but it does not particularly translate into what's going to happen in vivo or inside the body. So to do that, we wanted to further validate our docking studies in a more dynamic mimicry system. And here you can see the molecular dynamic simulation we conducted, where we took the, this is the cycloxanase 2, the best performing nervonic acid in the binding cavity. What you can see is that this fatty acid, there we can see it's rather jumping in a sense, but it's still stable in the binding cavity during the 100 nanoseconds MD simulation we've conducted. And this is translated when we look at the data as well. So first of all, the RMSD here, or the root mean square deviation, tells us about each of the amino acid residue of the protein and how they behave in the dynamic situation. So we can see that in the first 30 nanoseconds, it took some time for the drug and the protein to interact and be stabilized. After that, around 40 nanoseconds, reaches a stable equilibrium and diverges into a stable complex all the way up to around 90 nanoseconds, after which it starts to decline due to entropy. Uh, so here you can see the red line identifies the lipid protein one, and the green one is the drug protein interaction, and the green one is the protein by itself. So the RMS fluctuation is another metric we use to see how the drug and the protein is interacting. Here you can see that the conventional drug and the ligand molecule have a similar fluctuation pattern compared to the enzyme by itself. And another metric we use is the radius of gyration, and which tells us about the whole system and how it gyrates around the axis, or how it destabilizes around the axis. So our ideal drug protein complex would give us somewhat what we have here in case of the drug and the lipid protein complexes. So we're very happy with that as well. Similar data can be seen in case of the angiotensin converting enzyme, as well as for cyclophosphamase 2 and thromboxane synthase. So we have the docking results, which gave us an initial idea of the binding propensity of the ligand and the receptor in a static system, and the dynamic simulations gave us further validation of this interaction in a more in vivo system. So we took 100 snapshots of the entire trajectory, different time phases of the interaction, and tried to see the average binding affinity of the receptor to the drug-like compound, in this case, the fish fatty acids. And what we've seen is that in case of the hypertension-oriented proteins, that is the calcium channel as well as the angiotensin converting enzyme, the best performing acids and the best performing drugs had more or less similar energetics, although the drugs had a better electrostatic interaction rather than the hydrophobic and surface accessibility, which is the non-polar interaction of the protein and the ligand. However, this scenario is completely drastically changed when we consider more inflammatory routes, such as the cyclophosphamase 2 and the thromboxane synthase, where the lipid molecules seem to outperform the best drug molecule by quite a drastic margin in case of nucleic acid for cyclophosphamase 2, as well as oxidative nitric acid in thromboxane synthase. So from this, we can confirm the docking results in a more mimic system, and from that on, we took the top 12 docked lipids and simulated them into metabolic networks, or the KEGG metabolic pathways associated with atherosclerosis and inflammation. What's surprising here is that, contradictory to previous literature and so on, the compounds here were not very good at overall lipid metabolism and cardiovascular health. Rather, they seem to be better and more potent modulators of sodium and that is the apathetic caspase pathways, as well as the immune system, immune regulatory NF-kappa beta pathway, and aging and cancer pathways, rather than the conventional circulatory cardiovascular health related networks. So what we've done here is we've taken multiple protein checkpoints of these singular KEGG networks and simulated them against each of top 12 performing lipids. And the overall tendency here is that most of our high-performing fatty acids, you can see navonic acid here, triglycerolic acid here, octadecanoic acid, DPA, DHA, as well as linolenic acid, were mostly regulators of the caspase pathway and cancer-regulating pathways, that is the P53 and the STAT-related networks, rather than the lipid metabolism or the cardiovascular-related pathways. And if we zero in on the best performing singular molecules, we can see that navonic acid, dexahexaenoic acid, or DHA, dexpentanoic acid, or DPA, were the overall best performing compounds when we look at the vast, complex atherosclerotic molecular network in case of the drug and the biological pathways associated. So most of the points here I've already mentioned. We can see that most of the compounds we've considered were very good drug-like compounds, as well as we've exceeded 3 of the probable carcinogenic lipids from the study. Secondly, the blocking simulations revealed the initial tendency of the lipid molecules to be bound to the target receptors, as well as the binding affinities are mostly governed by hydrophobic interactions rather than electrostatic or hydrogen bonding, and the pro-inflammatory receptors we've mentioned here, which are the cyclooxygenase 2 and thromboxane synthase, were the ones that are targeted more propensely by the compounds. And in this case, navonic acid, DHA, and DPA were the best multiple pathway and multiple regulator modulating lipid molecules. And from there on, we went into a more dynamic simulation system where we saw that the compound receptor complexes were ultimately stable after 100 nanosecond production run, as well as we also went for the molecular mechanics calculation, which gave us an idea of the energetics involved during the actions. And finally, the metabolic kinetic simulations gave us an idea of how these lipid molecules interact with not just singular drug targets, but the entire atherosclerosis associated molecular network or metabolic network as a whole. And finally, we've, from a drug discovery perspective, we can see that navonic acid was the best drug-like compound if we consider atherosclerotic drugs by modulating aging, apoptosis, cancer, and cardiovascular diseases. And so for a minor take-home message here, we can see that mention some fish sources that have multiple of the best performing compounds we mentioned here, particularly tuna, the widely available fish species that has navonic acid, DHA, DDA, hexanoic acid, triglycerolic acid, and so on, and a very high overall polyunsaturated fatty acid content, followed by freshwater eel, limber, cod, catfish, and multiple oily fish species, particularly marine fish species. And with that, we can reach our conclusion. So from our study here, there's a lot of data and a lot of scientific terms, but the key take-home message here is that the atherosclerosis pathway that is complex and interconnected is actually mediated by multiple of these dietary fish fatty acids that we considered here, which gives us an initial idea of how these fatty acid supplements or fatty acid alternatives can help in heart health and circulation, as well as oxidation, cancer, and aging. And secondly, we've also gone for a in silico drug discovery route and collected navonic acid, decazopentanoic acid, DPA, and decazahexaenoic acid, DHA, to be not only very good multiple pathway regulators, but also substantially superior regulators to conventional drugs that are available right now. For further reading on the topic, our topic side very much recommend this and review on nutritional values of fish lipids by Straton et al., as well as some additional pages you may want to follow. And with that, I thank you for your time and your patience. I hope everyone's staying home and staying safe. And thank you.
Thanks a lot for this talk. Let's see. That was an excellent presentation. Let's see what are the questions. So, considering that so the lipids we were talking about, we already consumed them. So what are the challenges of looking at toxicity profiles? As we all know that the dose makes the poison, so how important it is to look at the dose that we are consuming?
Okay, so first of all, one thing is that once we consume them as food, because we're not particularly injecting them via intravenous injections, but rather as food supplements. So what happens is that most of our nutrients get degraded in the gastrointestinal tract, and then not much is actually metabolized. So that's why we have to conduct the in silico ADMET studies at first. So as you can see, in case of the gastrointestinal absorption, most of the compounds of the lipids we've considered had very good absorption ratios. This is important because most of the drugs we actually take don't tend to get absorbed or metabolized onto the cells as well as we intend them to. So this helps us gain a preliminary idea, and this is actually not very surprising since these compounds, as you can see, they're mostly hydrophilic in nature, which allows them to bypass most of the caspase cleavage enzymes in our gastrointestinal tract. They evade them and go into the bloodstream directly, and ultimately get easily absorbed as they are hydrophilic. And regarding toxicity profiles, we've went for the GSAR analysis and tried to predict how drug-like or toxic-like these compounds actually are. And all 32 of our considered compounds have very good drug-like properties. This can also be quantified. Is that in our study we took drug-likeness to be have a LD50 value or lethal dose value of greater than 2,000 milligram per kilogram. So each of these compounds have a greater acceptable dosage of 2,000 milligram per kilogram. Usually, this is a class 5 toxic profile. Usually, class 3 to 4 is considered mildly allergenic or mildly toxic, and 5 is safe to take orally, and 6 is completely safe to be administered.
I think you answered most of other questions I have, actually. There's some follow-up. There's some follow-up about possible future impacts of this study, and maybe it would be possible. Would it be possible to, or the good idea to have these lipids as introduced as individual supplements, for example?
I'm sorry, I didn't catch the last of this.
There is some feedback. I don't know.
Yeah, I'm also getting some echoes.
All right, so I guess we're going to stop
Can you hear me
now.
now?
Oh, there is some feedback. I don't know. All right. This was very interesting talk, and thank you very much for giving it. I will point everybody to ask more questions in YouTube and in Discord sessions, so we can know more despite the technical issues. And now,
and there's also a paper coming, so I hope I can answer more questions on the Discord channel, as well as for further details, the paper can answer.
Great, thanks a lot. All right. Okay, so now this is the end of this session, and I invite you all to a short coffee break around 15 minutes. Actually, it's going to be a bit longer, so around 20 minutes, I would say. And then during the coffee break, please go to the YouTube channel and go to the Discord and network if you can. And I would like to ask speakers in the next session to join our technical speakers channel so they can set up, and that maybe we can combat our technical difficulties together. Thank you very much. Thank you to all the speakers in this session.
this really small fraction of proteins that has been crystallized, we still have substantial amount of them to analyze, and you need software for this. So today I'm gonna present you a BioShell package that has been developed for a couple of years, actually several years now in my lab, that is devoted to analysis of biomacromolecular structures, mostly structures of proteins. And let me double check if it goes. Yes, so I will introduce you to the software just by showing how it's used. And the philosophy behind the package is that it's a suite of commands that you run in a command line, either on Linux or on a Mac. As you can here see, there is a program called app stacking interactions, and the program reads a PDB file and lists all possible or putative stacking interactions. And in the newest version of the package that has been just published this year, we greatly extended the set of commands, the set of programs. Now we follow the idea that one action, one function, one app. So we have like an explosion of applications and scripts that perform different calculations. And if you are, of course, the problem with such applications is that the possible functionalities that you can imagine is much more broader than the set of programs that we deliver. That's why we also deliver a Python library, so you can implement your own functions, your own functionality given the library. You just, if you are familiar with Python, you can easily recognize that we just import from BioShell, we import modules, and these modules, they then we call them, these are basically C++ objects. So you call them and run for what you want. This script, for example, converts multiple sequence alignment from class-style omega format to FASTA format. And of course, if we go deeper, we can use BioShell as a library for C++ programs. So this offers you the highest possible speed, but of course, for the price of much higher, much more steep learning curve and higher level of complications. But basically, these are the three ways how you can use BioShell package now: and command line, and Python scripts, and then C++ programs. And why Python? Well, I would argue that Python is the first language that you should learn, especially for young people, students who don't really know what future they will take, what they will do in their future professional lives. And so then Python is, well, it's very, very versatile and also easy to learn as a first language. So you can, in your lab, you can analyze data with Python, and when you go home, you can write a video game in Python. So you can do pretty much everything in that language. And as a second language in my lab, we use C++. So if there is a need for someone to write something that is really time consuming, then C++ is the way to go. In my lab, we also develop Rosetta software. We are actually Rosetta Commons member lab, so we actively develop Rosetta, which is in C++. So Rosetta Word uses basically the same tool set, Python and C++. And of course, we with this new third version of the package, we really improved documentation. So you can go to our Read the Docs website. I mean, our documentation website hosted on Read the Docs. This is automatically generated from sources. And you can find literally hundreds of examples, either Python scripts or C++ applications, and some cookbooks how to run command line utilities. Yeah, I think it's easy to approach. So this is BioShell from a practical perspective, how to run it. And from programmer's perspective, BioShell is a library of different modules. There is a calculation module, data in and out module, some alignments, some chemical module to perform actions on small molecules like ligands, for example, and algorithms that run all this data to calculate some results. So what are the features? What is the functionality? Well, first of all, it's data conversion. So we can read sequence formats like FASTA or PIR or CLUSTAL-W format ALN, different multiple sequence alignment formats. We can also read in and write structure formats. This includes PDB and CIF, but also small molecules formats like MOL2 and SDF that we use for docking. And we can also write some calculations with BioShell. For sequence, these are mostly alignments. We can align sequences, we can align sequence profiles. We can also do threading, which is alignment between sequence and a structure. So you can thread a sequence through a 3D template. And this is basically our tool set, our utilities set for comparative modeling that we use in our study. You can also run structure calculations with BioShell, and this is actually what it has been designed for. There are some simple properties like distances and angles, many of them. There are not so simple properties like molecular surfaces, interactions, RMSD, TM score. These are quality measures or distance measures or metrics or scores that tells you how two structures are similar to each other. You can calculate superimpositions and structure alignments. This is actually really used in my lab. Like now we have, let's say, 2 proteins or a set of P450 enzymes. Each of them has a heme cofactor, and we would like to superimpose these proteins by the cofactor. So unlike all other superimpositions, when you typically superimpose alpha carbons, we want to superimpose heme as good as possible. And then from this superimposition of 2 heme molecules, we calculate rotation and translation. And then we apply this rotation and translation to C alphas. So C alphas are superimposed by heme. And there is an easy script in BioShell. And BioShell itself provides routines to calculate this rototranslation, and a script, you just tell what to be rotated. So this is the workflow we do. There are also other features, all other functionality. We often use hierarchical clustering. When we have models from molecular modeling, from docking, or from other means, we can also cluster sequences. And we also do some statistical and numerical analysis with the package. And this is like a graphical overview or graphical set of examples that we can do. We can, as I said, we can calculate any distance and angles. Having the ability of computing distances and angles, we can detect interactions like hydrogen bonds in the top middle or stacking interactions on the top right. These are interactions detected by BioShell, and the way it goes, as I already told you, we run it in a terminal or in a script. So this way we can massively process lots and lots of PDB files. So the bottom row shows that we can gather statistics from whole PDB, or for example, 1 million poses from docking computed with Rosetta. And yeah, we run it on a cluster. And we can also calculate contact maps, hydrogen bond maps, and so on. So these were pretty easy and oftenly used applications, oftenly used statistics that you calculate from structures. And there are pretty many software packages that can do this for you. Probably the most known and the direct competitor of BioShell is BioPython. So yes, you can do the same with BioPython probably. Maybe a bit slower because it's Python, BioShell is written in C++, and in Python you only call C++ routines. So PDB parsing, for example, is done in C++ layer. And there are also more, I would say, advanced applications of the package that are not that easy to find on the web. And this one that I'm gonna show you today, it's something that we started when I was actually working at the University of Virginia in Charlottesville. And then I was a postdoc in a crystallography lab, and we got this crystal structure, which you see already now. It looks like any other protein. I mean, nothing special, yes? And when you actually inspect it closer, you will see that the middle beta sheet, it's composed of a few strands coming from two different chains. So you see a pattern of green and red beta strands within a sheet. For me, it was actually interesting how the protein folds. And we call this protein interdigitated. So it was first published in 2009. So now, given the new version of BioShell, I decided to check what's new in PDB, if there are any new deposits that have this feature. And I asked, I assigned this to my student, that this will be his graduate project. So BioShell can parse the PDB as a graph of secondary structural elements. And in this graph, this is just a picture of a random graph. But in our case, strands are nodes of the graph, and hydrogen bonds between strands are edges of the graph. And then you color the graph, just you would color the chains in PDB. So some nodes of the graph are red and the other nodes are green. And then you look for the path that goes through alternating nodes. So nodes of alternating color. And in this particular case, you see AB, AB, AB pattern. So six chains. So along the hydrogen bond network, within a given beta sheet, the chain symbol is changed 6 times. And actually, we found 6 such deposits. 2 of them were known in 2008 when we first published the paper. And now we have 4 new interdigitated proteins. So this is a very fresh application of BioShell that is actually going on now in my lab. So this is not published data. So yeah, so we have some plans for the future, of course, for the package. We are pretty much ready with binary distribution. So far, you should actually download the source from Git repository and compile it. And this runs pretty well on every Linux and Mac machine. With Windows, it's a bit problem, so we still fight with this. And I understand that this terminal thing or Python programming is not for everyone, and we would like to extend the group of users. We would like to approach also users who don't do programming at all. So that's why we try to, the newest feature. In the newest future, my lab will deliver web-based applications, which actually are written in Python as well. So Python is very, really handy. And for this, we wrote a visualization library in Python as well. It uses so-called Brighton. It's a Python version that runs in a browser on a client side. Also on the server side, we also use Python. So now I try to do something really hard, and on the fly, I try to unshare this screen and share another one, which is this. So is it okay? Can you see it?
Yes, we can see it. Thank you very much.
So it went okay. Yeah, so here is a web application that presents results from docking. The target was actually, the receptor was P450 enzyme. We actually had 12 orthologs, so 12 sequences coming from different bacteria and fungi. And we had 8 drugs. So every square here represents one combination, drug versus ortholog of CYP51 enzyme. And for each case, we generated 10,000 decoys, 10,000 docking poses. So it's over 1 million of structures. So the data takes about 2 terabytes on a hard drive. So it was really, really practically impossible to browse the data to find something interesting to put in the paper, actually, you know. So then I decided to write this application, as I said, in Python. And you can, for example, check models, and you can click on a single box. There's an energy, Rosetta score versus some quality check, some distance measure, and you can browse for positive and negative results. As I said, this is the docking has been already done. This is just a browser to the data written in Python with BioShell. BioShell actually works on a server, and it loads PDBs and gathers statistics and sends the statistics to the browser. And browser just displays them. And in the future, we will extend this application with docking capabilities. So Rosetta will do docking on the fly. We'll just be browsing and docking at the same time. So we use PyRosetta. PyRosetta is a Rosetta molecular modeling package, also in Python. The same is as we did with BioShell, which means that you call C++ from Python. Okay, now again, a difficult moment. Yeah, I would like to go back to my presentation, which apparently I don't know how.
Can you try maybe switching between windows with Alt
Yes,
Tab?
yes, I tried to share another screen, basically. Okay. I tried to share another screen, but okay, so I can basically tell you what's on the final screen, because on the final screen were just acknowledgements, and yeah, but maybe yeah, that's why I was so fast with my presentation, you know, because I assumed that something will go wrong. And
I mean, that's a 2020 in a nutshell, I guess. Maybe you can close this one, this window, and then that one will appear. Hopefully.
Okay.
Let's see. I'm still seeing the docking window. No, nothing.
I'm selecting a screen to share now.
Awesome, awesome. Still have time, so it's gonna be okay. It's gonna we're gonna edit it out after to leave it on YouTube with a edited version, so no one will notice any issues.
Good.
Did you share the screen, or is it still?
Yes, I now know what.
Okay. Okay.
So can you see the screen, the presentation now?
I can't, but hopefully our IT people can.
It's shared. I mean, it's being shared now.
I guess it's another LBS session.
Okay.
Well, I guess I can use this moment to say a couple of words, because I hear that we had some interruption at the beginning and no one heard what I said at the beginning. So I just want to repeat a couple of things. So I'm your chairperson. My name is Kasia Kędzierska and I'm a PhD student at Oxford University. We're hearing the talk by Dominik Grond from University of Warsaw on BioShell, a versatile package for structural bioinformatics. And we're waiting for the presentation to appear. Hopefully it's going to happen any second now. Let me check that.
It's already there.
Okay, so I hear from our team that we only have your web camera.
And that's it.
Yes. So maybe if you can just proceed as it was there, and then we're going to edit it out when you send us the slide. We can just paste it there.
Actually, I can send you the PDF file with the presentation. I can share it, so it's no problem. And I expected actually some troubles with this, so I decided to do this switch between windows on the very end of the presentation.
Mm-hmm. Of course, that's a little bit of technical issues, yeah, always.
So the very last slide basically shows acknowledgements. So I would like to thank my students that developed the package and tested the package. Actually, the most of the time that you have to spend on a software project is to error checking and debugging. So just coding is easy, and testing is very time consuming. So in the last paper, with BioShell paper, we introduced a set of tests. So there were students who developed these tests in Python and in C++, and there were also students who contributed C++ code. So Ola, Aleksandra Balaczewska-Dawid contributed to the package. Joanna Matznar and Justyna Kryś are now my two PhD students, and they developed the package. Joanna is doing docking, so the results that I showed you with this massive docking experiment was done by Jasia. And Justyna is developing BioShell in C++, manages the package, and also starts developing Visualize library. Visualize library, I recall it's this Python library that works in a browser to allow for Python-style visualization. I think it's the only the only library in existence that does it. Everything else, every other library works in JavaScript. And Natalia Schulz helped us. It was actually her master project to bring the set of biologically relevant examples. And also I have to acknowledge Mateusz Kłodowski, who did this interdigitated proteins project. And that's all from me, and I'm ready to take questions. I hope you have some interesting applications for the package that I could help you with. Thanks.
Thank you very much for your great talk. I have a couple of questions. So I'll start with a question from Rahagir. So he says, thank you, Dr. Grond, for your informative and intriguing presentation. Sorry for being an amateur, but could you please point out the strengths and advantages of the package over available tools or platforms?
So when I get emails from users that would like to, and what they see as a unique feature is ability to calculate TM score and GDT scores that are used in molecular modeling field, especially comparative modeling, and that are not that easy. That code is not that easy to find. Also, the speed. I mean, we compared with BioPython. We have not that many functionality related to protein sequence. So, for example, BioPython can directly access a sequence from a gene bank and align it. We cannot. We just read in FASTA files that you give to the user, that user gives to the package. And but the advantage is that everything in BioShell is written in C++ and not in Python. So you have to compile it. But once you compile it, you get like an order of magnitude faster in some applications.
So actually, regarding the compilation step, we have a question. Will BioShell be available as a Docker container, as it sometimes is quite cumbersome to compile it from source?
Yes, that's definitely on our list. Actually, Justina is already working on that, and she told me that she already has a version, and so as a Docker is easy to get this as a Docker, and we already have this. We had even more ambitious goal. We would like to compile within Docker, so we can share binary files to everyone. Yeah, but compilation within Docker is more difficult than just installation within Docker. So yeah, we can share these Docker components. Compile.
I guess you already touched on that, but do you predict possible integration of BioShell into other common software used in the structural biology field, where there are a lot of users that are more used to point-click interfaces?
Yes, I now with this tool set already developed, by tool set I mean this Python library and C++ library, which these are pretty matured. Now the development goes towards web application, and so I would imagine such a use scenario that if there is a laboratory that has some computational resources, an admin or a PhD student or someone really skillful can install applications on the server, and the whole lab can locally access them and run jobs on the local cluster. We do this with Rosetta, for example, and BioShell. So BioShell is used to analyze outputs from Rosetta, and you can do this through your browser. Of course, you can do this from home. I mean, what my point is that I don't have enough resources to share all the functionality for the whole world. But I can deliver you a graphic user interface that you can install in your own lab, providing that you have some Linux boxes. Okay,
and I have one question. Sorry if it's too broad or too basic, but I'm mostly a person who works with genetics, genomics data. But when I was looking at your presentation, I kind of saw an application in cancer, because what we usually do, we deal with somatic mutations in the coding regions. So I would be interested if it is possible to see where my possible somatic mutation is in the structure of the protein. Do you think I can visualize it somehow and see maybe the mutations that are related to the binding site, or related to the like whether they are inside or outside of the folded protein?
So if you have structure templates for these proteins, then it would be a really interesting project to make such a browser. By a browser, I mean a web application that you will put in some genetic data plus the templates, and then there will be ability to see where the mutations are located in the protein. If you don't have structural templates for these proteins, then I would say it's hard. But for many proteins, we have templates. Like structural genomics delivered templates for majority of PFAM families. So unless your proteins are orphans, then I would say it's highly probable that there is a template for modeling for each protein. So
Thank
yeah,
you.
let's keep in touch. This is really interesting.
Thank you very much for your interesting talk, and I guess I will be looking at BioShell later then. Thank you very much, and I guess we are going to go to our next keynote speaker, if I'm hearing that correctly.
So should I switch off these shares?
Yes, if we can ask you to do that. I guess we're having a little bit of technical issues. We cannot see the next speaker yet. So give us like 2 more seconds to figure out what's happening. Okay, guys. Okay, people. So what we're going to do right now is, because we're having slight technical issues. Oh, hi, Rosa. Can you hear me?
Hi. Yeah, I can.
Okay, great. Did you share the screen of via OBS?
Yes.
Okay, great. So.
Can you see my screen, or?
Yes, perfect. We're gonna go with the plan then. Okay, it is my pleasure to introduce the last keynote of the Virtual NGS Symposium 2020, Rosa Tormo. Rosa leads a research group at the Wellcome Sanger Institute, focusing on immunogenomics, reproductive atlas, and cellular networks. Rosa did her PhD in Barcelona, where she studied the influence of cytokines on innate immune cell differentiation. Later, she did her postdoc. She did her postdoc with Sarah Teichmann at the Wellcome Sanger Institute. There, she worked on tissue-resident immune responses and focused on one of the most intriguing environments for immunologists, the maternal-fetal interface during pregnancy. Rosa also designed CellPhoneDB, a novel repository for ligands and receptors and their interactions. Today, she will give a talk about temporal and spatial dynamics of human endometrium. Rosa, thank you very much for taking the time to be here with us. And without further ado, the floor is yours.
Okay, thank you very much for the nice introduction. And yeah, apologizes for the technicalities that we have had here. So, well, as you said, our team uses single cell and spatial methods to deconstruct tissues. And in this way, we can learn about the mechanisms and heterogeneity.
I'm really sorry, to interrupt you, but we can't see your screen at the moment.
You can't?
No, it went...
Okay, you cannot see my screen?
No, I think our technical team cannot see this share. Maybe the OBS actually crashed.
Okay. I don't know how to share the screen because I already shared. So if you tell me what to do.
So please go once again through this OBS link and try to share the screen in a new tab in your web browser.
Yeah. So Carlos. Okay. So sorry. What do you want to do?
Please produce a new tab in your browser. Go to this OBS link, similar one to this you used to share your webcam. When you see the choice between the web camera sharing and screen sharing, choose the screen sharing one and then choose the window or the screen with your presentation.
Screen share, right? Which one? No, no. Screen share. Yeah, application window. Yeah. Okay. Is now
okay? Yes, we can see it.
Ahora ves el PDF en PowerPoint. No toques eso. Okay. Ahora ponlo en tope. Yeah. Okay, now can you see it?
Yes, we can.
Okay.
Thanks.
Okay, yeah.
Can hear my voice? Yeah, can you see my screen?
I don't think we can. I think it's when you click the present in Microsoft PowerPoint, it disappears because it
Okay,
sees a different window.
now you cannot see it. Now
Yeah.
can you see it?
No, still not.
Okay. I really don't understand because I'm sharing my screen. So
maybe you can try actually clicking present and then sharing your screen and choosing this screen as the one to share. Well, I missed the issues we had like earlier, like finding the cable to connect to the projector, because this is the equivalent of that, I think.
Okay, so now I should go here. With room, right?
Comparte toda la pantalla.
But I cannot find my okay now. Okay. Now the source. Now can you see my screen or?
Yes, okay.
And now?
Still it's visible. Okay, it's fine.
Can you see my screen?
Yes.
Yes, it's fine now.
Yeah. Yes.
Okay.
Great. Okay. I'll let you get to it.
Yeah. Okay. Sorry about that. Well, anyways, I was saying that we use single cell spatial and transcriptomics methods to deconstruct the tissues, and in this way we can learn about the mechanisms and heterogeneity of the tissues. And then we also use in vitro models such as organoids to dig more into these dynamics and the mechanisms. And actually something that we are quite excited now is in these in vitro and in vivo comparisons that are allowing us to make more realistic in vitro models. So in the lab, we have two main areas of interest. One is related to mucosal immunity, and the other one is related to early development. And for this talk, I'm going to focus on this early development one. So instead of focusing on the embryo itself, I'm going to focus on the environment that makes possible this early development. And this is the uterus, and in particular this endometrium, which is the mucosal layer of the uterus. That, as you can see in the histology here, is comprised by these epithelial glands, these invaginations of the tissues, and also this epithelial tissue in the surface that will allow the embryo to attach. So altogether, this endometrium, by either helping the embryo to attach to the uterus or also by providing nutrients that are stored in these glands or invaginations, allows this embryo to develop. But it's very little we know about the endometrium, also because of their challenges. It's quite a dynamic tissue. So as you can see in this slide, the endometrium goes to monthly cycles of regenerations. So it goes, it sheds during the menstrual cycle, and then it regenerates, and after ovulation, it differentiates. And this cycling process is regulated by by two hormones, estrogen and progesterone, that are produced by the ovaries. So another challenge for this tissue, and one of the reasons why we know very little about it, is because these cycles of regeneration and differentiation are quite unique to simian primates. So in mice, for example, the full differentiation do not occur until the embryo is attached, while in humans, this differentiation occurs independently of embryo. So due to the technological and developments, we are now in a position to be able to study this human tissue. And that's quite exciting because there are a lot of pathologies associated to this tissue, not only infertility, but also some others, for example, endometriosis, which causes real pain in 1 out of 10 women in their reproductive age. And due to the delay of what we know about this tissue, there is still no diagnostic tools or either treatment for those women. So the main technological advances that are making now possible to study human tissue are all these single-cell atlasing methods, and this includes single-cell transcriptomics and all modalities, for example, open chromatin, and also all the spatial methods. And for example, in this slide, I'm showing how during my postdoc with Sarah, I used single-cell transcriptomics to identify the heterogeneity of this maternal fetal interface during pregnancy, and that allowed us to characterize novel immune populations and how they communicate with the fetal cells, and also this new population of stromal cells that you can see here in different colors, and by combining them with spatial methods, we managed to give a special position for those new populations. So on top of that, there is all these organoid models that are allowing us to study the development and differentiation of the tissues. And in particular here, In 2017, our collaborator Margarita Turco developed these endometrial organoids that recapitulates epithelial differentiation in the endometrium. And actually, they are quite unique because they are able to respond to the ovarian hormones as it happens in vivo. So with all of this, we have a lot of questions we want to resolve, but we are going to start very simple. First of all, we want to understand what are the cell types that make up the endometrium, what are their locations and how they change throughout the cycles. Then we also want to know if the organoids available recapitulate epithelial cell heterogeneity and if there are ways to improve those models. And finally, if we have a good model, then we want to understand or dig more into the key signaling pathways that control this endometrial proliferation and differentiation. As I was mentioning before, our study will have a direct impact in women's health, such as endometriosis, which are really diseases that have been neglected so far, but also for basic or fundamental questions such as regeneration, because this tissue has the capacity to self-regenerate itself in a monthly basis. So in our experimental design, we combined two types of samples. So on the first instant, we combined biopsies, which only take the superficial layer of the endometrium. But also we included organ donor samples. So those are full uterus from healthy, people who died but gives their organs for donation, and that really allows us to look at the full thickness of the uterus with all the layers, including the basal layer, which is the layer that sticks to the uterus and most likely the layer that contains the stem cells. So what we did is to sample different individuals, and in this way we can cover the different stages of the menstrual cycle. And yeah, we combine single cell transcriptomics methods with some special methods such as Visium or RNA-scope, and in this way we can give a spatial coordinate for the new cell types identified and start digging more into the cell-cell communication. So to start with, we perform this single cell transcriptomics, and these reveal the novel cell types we find in this tissue, which includes novel populations of fibroblasts cells and other endothelial cells and perivascular cells around the endothelial. And here we can see in this plot how some of them will come only from the biopsies, while others came from both the biopsies and the organ donor program, just highlighting how relevant it is to have these samples in our study. So then we have all the distinct cell types, but then we also want to know the spatial coordinates. And to do so, we perform Visium analysis. So basically in this analysis, you have a tissue slide where you will put the tissue and image the tissue. This is just an H&E of the different tissues in different stages. But what is unique about these slides is that they contain these areas with barcoded spots, and these barcoded spots contains the poly-DT that will bind the RNA that will diffuse from the tissue to the slide, but also this special barcode that will allow in the future to match the RNA to the image that you perform here. So one of the limitations for this method is that it's not single cell resolve. you have 50 micron areas. So then these 50 microns is between 10 or 50 cells, depending on the tissue. So what we did to overcome that is to kind of integrate both the single cell transcriptomic data with the spatial data from the same donor, and in this way we can give an estimate of the proportions of cells that you have in each of the capture areas. And to do that, we use a new method that is called cell-to locations. That it's basically this probabilistic method that will infer the proportion of cells in the areas. So after this, then we can assign what cell types came mostly from the endometrium in blue. So this area that will regenerate monthly, and the one that comes from the myometrium that is mostly a smooth muscle layer. So we not only confirm what we know, so for example, we know that the smooth muscle cells are in the myometrium and that immune cells are more in the endometrium, but also this allows us to give novel identities to the populations we find. So for example, in our single cell data, we find this Fibro2 population, and now we can see that actually this Fibro2 population is in the basal layer within the endometrium and the myometrium, and then we can give this fibroblast basal identity to this new population. So then we were mostly interested on the epithelial fraction, because this is the epithelial cells in this tissue are the main the main functional cell type. So because it really allows the implantation and provide the nutrition of the embryo. So in the differentiated tissue, we have mainly two cell types: the ciliated cells that are in the lumen and that will spread out the secretions, and then the secretory cells that are in the glands and that will provide the secretions that will be key for the nutrition of the embryo. So all these cell types regenerate from progenitors every month. And actually, when we cluster our different cell types, we have find the secretory and ciliated that are the differentiated cells on the stages of the menstrual cycle that correspond to the secretory stage. But then in the most proliferative stage, we find different intermediates. So this is just to show you how then we combine the markers we identify by single cell transcriptomics with single molecule FISH, and this really allows us to characterize how these populations change over the menstrual cycle, and how this PAAP, which is a differentiation marker, appears after this secretory stage. And how this NNP7 is mostly expressed in early proliferative stages. So after characterizing the main populations, we wanted to dig more into these proliferative phase subsets, these intermediaries ones. So we reclustered them, and we found some clusters that are responding to the progesterone hormone. So they have high levels of the progesterone receptors, and some others that have lower levels of them. And what is interesting is that from the ones that have high levels of this progesterone receptor, we find this LGR5 population, which express high levels of the Wnt pathway, and other markers that are expressed on other progenitors from other tissues like the lung. And then actually what we find is that this LGR5 population that is also expressing with 7A is highly expressed in the early stages of proliferation, that is these menstrual stages, that is when all the revitalization is happening. So that really made us think that this is a novel progenitor that we find in our tissue. So then we wanted to know more about how the different microenvironments are regulated. So we know that in this tissue we have the lumen, and then we also have the glands, and we know that different microenvironments after the differentiation enrich on different populations of epithelial cells, and our question is how this microenvironment help for the differentiation of epithelial cells go through or enrich on the ciliated lineage or the secretory lineage. So to do so, we took advantage of the spatial transcriptomics data, and what we did is to do unbiased clustering. And by unbiased clustering, then we selected the cluster corresponding to the epithelial subset that we then reclustered again. And what we find is that nicely, this unbiased clustering is recapitulating the different microenvironments. So in yellow, we can see the lumen, the epithelial lumen, and in blue, we can see the secretory glands. And then in red, we have the basal gland. And actually, when we look for the markers of each of the clusters, we can see how in the lumen we have markers for this LGR5 population, and also this FOXD1, which is the ciliated populations, which match with our prediction for this for a single molecule fish. While in the glands, we have markers for the secretory populations, and these clue ones in the basal layer, as we predicted before. So in addition of those markers, what we found is that we also find in the lumen some genes that correspond to the Wnt pathway, such as Wnt5A or Wnt7A that we mentioned before. While in the gland, we have some genes that downregulate Wnt pathway like AXIN1, and also some NOT genes like NOT2. So these really point towards an enrichment of the Wnt activatory signals in the lumen, while those are reduced in the glands, and the opposite for NOTs. So we wanted to look more into these pathways. And to do that, to measure this activity of the pathways, what we did is to measure the activity of the transcription factors in the different environments. And to do that, we use Dorothea. So what we find is that in the lumen, we have transcription factors that are active in a Wnt, are active by Wnt, while in the gland, we have transcription factors that are active when you have a Wnt inhibitor environment, and also some transcription factors that are active by NOT. So this really matches again with the D expression genes, just suggesting that we have a higher Wnt activity in the lumen and a higher NOT activity in the glands. So we wanted to dig more into this regulation of NOTs and Wnt in the different microenvironments. And to do that, what we did is to use high resolution imaging, and we quantify the signal of Wnt and NOT in the glands and the lumen. So here we can see how some members of the NOT pathway, such as NOT 1 and 2, go down into the lumen, as our single cell data and spatial transcriptomics data suggested, while some of the members of the Wnt pathway, such as Wnt9 and Wnt7, go up in the lumen. So, okay, there is this balance between Wnt and NOT in the different microenvironments, but then we wanted to know how other cells contribute to that. So epithelial cells are embedded in the tissue, and in the tissue we have these specialized fibroblasts that are called endometrial stromal cells. So those endometrial stromal cells also respond to hormones and also proliferate and then differentiate. So the first thing that we did is to reconstruct this cycle of regeneration and differentiation. And indeed, yeah, we can find all genes that have been described supporting this trajectory, and also donors who are proliferating are in their proliferative stage, and the ones that are decidualized or more differentiated are in the later stages of the menstrual cycle. So to study how these stromal cells communicate with the epithelial cells, we took advantage of our method called self-on-DB. So self-on-DB is a repository of ligands, receptors, and their interactions. And what is unique about this database is, first, that it's a curated database. So we always store the PubMed ID associated to these interactions, but also that considers that the majority of receptors and ligands are multimers. So you need more than one subunit to be expressed in order for this receptor or ligand to be functional. And that's important. And actually, the majority of ligand receptors in databases do not consider this. And while that's important for a lot of pathways, and here just showing you how, for example, in the Wnt pathway, you really need the expression of several subunits in order for Wnt to bind and have an effect on the cell. And that applies both for their activation or their inhibition. So in our previous work, what we did for self-on-DB is to integrate it with single-cell transcriptomic data and to look for ligands and receptors that were specific for two cell types. And to do that, what we did is to do random shuffling of the ligands and receptors in the single-cell data set. However, these specific data set that I'm presenting now, it's more complex because it changes across time and space. But the good thing is that we have this information about time because we have the different clusters on the single cell data, and we also have the spatial information. So what we did now is to modify slightly the method for self-on-DB, and then we, on top of considering the expression of the ligands and receptors that are specific for cell types, we also consider those that colocalize in time. So if the cell types are in different times, we don't consider them in the same model. And then also we consider cells that colocalize in space. So if a cell is in the lumen and another cell is in the glands, we consider that those cells are not interacting. So modifying this self-on-DB now, we got the results, and here I'm just plotting the results for Wnt and not. And basically on the diagrams on the top, we have the proliferative phase, and on the bottom we have the secretory phase. And then in the proliferative phase, well, the different colors represent the different spatial microenvironments. And then we can see how in the top panels, we see activity of Wnt kind of everywhere expressed, just highlighting how important Wnt is for maintaining the proliferative stage. However, when we go to the secretory stage, we see how fibroblasts that are decidualized start expressing DKK1, which is an inhibitor of Wnt, and this is only on the decidualized fibroblasts that are close to the glands but not to the lumen. But also we see how these Wnt receptors are downregulated in the epithelial cells in the glands, the secretory cells, but not in the ciliated ones that are in the surface. So that really suggests how it's not only the regulation by the epithelial cells themselves, but also how stromal cells also contribute to this inhibition of the Wnt pathway. So altogether, for this first bit of the talk, what we have been doing is to use single cell methods to generate a map of the endometrial differentiation, regeneration, and differentiation, and also to be able to assign the different pathways that are playing a role in this epithelial differentiation towards the ciliated lineage in the lumen or the secretory one in the glands. But yeah, one may ask, like, why is this important to do such a map, right? And well, we think that those maps really are quite relevant in the sense that you can, if you have a healthy reference, then you can inform what's happening in disease, and also by comparing it, and also by helping to kind of design more realistic in vitro models that will then also help you to dissect better the mechanisms. And this is where I go with my second bit of the talk. So to dig more into this and also to try improve the current models, in vitro models, then we started with the endometrial organoids, which are these, those are primary derived cells that you just put in a dish and matrigel, and then you get the organoids. And what is quite interesting is that after adding hormones to this in vitro culture, you can differentiate them towards the secretory lineage or the ciliated lineage. And this bit that I'm going to discuss here was performed together with our collaboration, Margarita Turco in Cambridge University. So the question we ask here is how well this endometrial organoid system mimic the endometrial epithelium. And second, if we can functionally evaluate the signaling pathways that we identify from the in vitro system. So in our experimental design, what we did is do perform a time course where we profiled both the proliferative stage, which in vitro is just recapitulated by the addition of estrogen, and the secretory stage when we add estrogen and progesterone in the media. And we combined 4 clonal organoids from 3 individuals. So so what we see after we perform the time course is how after we add estrogen, then we have these intermediates that start expressing the PGR receptors as it happens in vivo, and how they then differentiate into the main lineages, the secretory lineage and the ciliated. So by comparing markers, both in vivo and in vitro seems to be quite consistent. However, we wanted to have a more quantitative assessment of these in vitro and in vivo comparisons, because if we have this quantitative assessment, then we can also see if we improve the media, we can say, okay, we are improving or not improving in a specific number, right? So what we did is to use logistic regression to compare both in vivo and in vitro data. And this really gave us a specific p-value of how this data is compared. And on ciliated, we can see how the differentiated populations, ciliated and secretory, both have a good match from when we compare the organoids with the in vivo. And then also in the intermediate stage, we can also see a good match with all the proliferative samples that we see in vitro, and also the ones that are starting to express the PGR receptor. So then we have a good model and we are happy with it. And then what we decided to do is to use this model to inhibit nodes and wind and see if this has an effect on the ciliated and secretory lineages we predicted in our in vivo data. So here we just did a bulk assignment. So we just did quantitative PCR for markers for ciliated and secretory. And quite exciting, we see that actually if we inhibit NOT, then we have an increase of the ciliated markers, suggesting that this NOT inhibition promotes the ciliated lineage, while if we inhibit Wnt, we have the opposite. We have an increase of the secretory cells and the ciliated are just decreased. So just again highlighting this balance of Wnt and NOT and the effect on epithelial identity of the endometrium. So we did the same, just analyzing the protein level by immunohistochemistry, and again we can see how NOT inhibitor is really increasing the ciliated populations and decreasing the secretions, and the opposite effect is found when we inhibit Wnt. So we wanted to have a more quantitative assessment of this, and also to see if are we just creating more ciliated cells, or are we our secretory, or are we improving those ciliated secretions? And to do that, we perform a single-cell transcriptomics analysis. So what we find is that for the ciliated populations, as you can see here, we have a significant increase of them when we have NOT inhibitor, while this is while the secretory populations decreases, and the opposite pattern is happening for Wnt inhibition. But what is also quite exciting is that actually when we put Wnt inhibitor, what we see is a new secretory population. And when we assess this new secretory population, it seems to be expressing more PAP, so more likely to be more realistic and more similar to what we find in vivo. So again, to assess if this is true, if the population that we are having in the secretory is more realistic than the one that we have when we don't put Wnt inhibitor, we did a logistic regression, and here we are projecting again the in vitro signatures into the in vivo epithelial data set. And actually what we find is that these new population of secretory cells are more similar to the in vivo populations, just suggesting that we are creating more realistic secretory populations by inhibiting Wnt. So just to mention that we also try do the same without hormones, and what we find is that if you don't have hormones, then you still have an induction of secretions, but it's quite poor. It's not as high as when you don't put the hormones. So basically, there is an addition effect and an interaction between the hormones and the Wnt and NOT inhibitions. So then we wanted to know more about the molecular regulation and the transcription factors playing a role during the identity of ciliated and secretory cells. And to do that, we perform single-cell ataxic. And again, we perform the single-cell ataxic in different time points. And we transfer labels from the single-cell data into that single-cell ataxic data. So it was not the same cell, but it came from the same suspension. So what we did from the transcription factors is to look for the accessibility of the transcription factor motifs, and also the accessibility of genes that encoded transcription factors. And then what we also did is to integrate 4 measures. So 2 for the transcription factors that I mentioned before, and also at the RNA level, the transcription factor activities and the transcription factors that are differentially expressed. And we compare these for the in vivo and in vitro populations. And what we find is that in the new secretory population that appears when Wnt is inhibited, we actually have a lot of genes that are activated by NOTs. Also, just highlighting how NOTs and Wnt also talk to each other during this differentiation by having an antagonistic effect. And actually, we also see some of the Wnt inhibitor and transcription factors as we will expect because we are inhibiting Wnt. And on the ciliated case, we have the opposite. We have they appear or they are more efficient when you have NOTs inhibition, but then you have a lot of transcription factors that are known to be regulated by Wnt. And these transcription factors are common both in vivo and in vitro. So it's quite exciting to use these technologies to disentangling all the specific mechanisms. So to sum this bit, what we have done is to use single cell methods to map in the organoid differentiation and to dig more into this pathway of epithelial specification. And we find how NOTs inhibition is key for to obtain ciliated organoids, and how Wnt inhibition is key to get more secretory organoids, and actually those secretory organoids are more realistic, and how these factors that we find in vitro are also comparable for what is happening in vivo. So then we also wanted to know what if these Wnt signaling alterations can also be happening in endometrial pathogenesis. And so far we only have healthy samples, but what we did is to just use some GWAS hits and see what are the cells expressing the genes that are found in the GWAS. And actually what we find is that a lot of them are expressed by the epithelial cells, and that's expected because those are the main functional players of the tissue. But actually we also see that some of the genes are expressed by fibroblasts. And also what we find is that a lot of them are secretory molecules that are involved in the dialogue between the fibroblast and the epithelial. And actually we find that a lot of them are involved in this Wnt dysregulation. So basically for this bit, we find that this Wnt dysregulation can potentially be involved in these endometrial pathogenesis, such as endometriosis and endometrial cancer, by the dysregulation of the Wnt ligand. So altogether, to summarize our talk, so basically we have built this atlas of this highly dynamic tissue. And we have gone further and also described the mechanisms existent on each of the microenvironment, this Wnt and NOT, this regulation in the glands and the surface. And then we have also used the organoids to dig more into how these pathways really have an effect, a causal effect on the identity of the epithelial cells. And then we have found how these pathways can play a role on endometrial pathogenesis, just highlighting how relevant studying them is. And just to finish, I would really like to thank my lab, and in particular Luz Garcia Alonso and Louis-Francois Hanfield, who are two postdocs, computational postdocs, who have been working in this project and optimizing all the in vitro and vivo comparisons. And then I also would like to thank my collaborators, Margarita Turco from Cambridge University for all help with the organoids, and Bayraktar team at the Sanger Institute for help on imaging, and all other teams and donors. Thank you.
Thank you very much for an amazing talk. I am a little bit biased doing a PhD in endometrial cancer, so I find your talk extremely useful. So thank you very much. I have a couple of questions, but I'll start with the ones that were asked by others. So Mystic Mike asks, I think it was around the time you were talking about the organoids, are these biopsies taken from pregnant women?
No. No, the biopsies are because we are just studying more the dynamics of the endometrium. So they are just taken from endometrium. So it's actually women who go to the clinics because the partner has male infertility. So they are fine. Yeah.
So I also had a question about the age of the women that you took biopsies or the uterus from. What's the age of your donor?
Yeah, so we included only women in the reproductive age. So it was between 25, I think, and 40. Yeah. And both for the organoids and the in vivo data.
Okay, so my other question would be, because you mentioned that you looked into what can be done with the data that you generated into endometriosis and endometrial cancer. Did you look maybe at the epigenetic changes, so like transcription factors or like chromatin remodelers that are being expressed throughout the menstrual cycle? I am asking this because my interest is in, obviously I'm being very selfish here, but my interest lies in chromatin remodelers, and those have been linked as highly mutated both in endometriosis and in endometrial cancer. And I would be curious to know whether you see some kind of changes in expression of those proteins throughout the menstrual cycle.
Yeah, so for now we have only generated healthy data, so it's not. So we only took for the diseases we only took the GWAS hit. So yeah, we didn't look at this. We are generating now. So I'm afraid I cannot respond to this because in the GWAS hits there were not that many transcription factors. Yeah, so I'm sure they will come up, but yeah, we don't have this data yet.
Yes, I'll rephrase my question because I think I didn't pose it properly. Because I'm asking whether when you look at the temporal changes in the menstrual cycle in the expression data, did you see a lot of change in some transcription factors activity of expression of the transcription factor or chromatin remodelers?
Oh, yeah. Sorry. I thought you were mentioning about the disease stage. You mean in healthy? Oh, yes. Yeah, we see a lot of transcription factors. There are big lists. Because in our first question, we were more interested on what is driving the identity of the epithelial cells. We just focus on those. But yeah, there are a lot of transcription factors changing across the dynamics of the tissue. Yes.
And we have another question. I guess it's related a little bit to the earlier talk we also had about the ethnicities. So would there be any differences between ethnicities if we would look at this data from different participants? And I guess a question related to this, did you have different ethnicities among the samples that you've analyzed?
Yeah, so I think there is always a question when one do these atlases on if you go more into depth or you just expand to a more population study. So in this first study, we decided to go more in depth for few donors, and for that we just consider one ethnicity. Saying that we are expanding now the collection, so we are part of a European grant. It's called UTER, where we are sampling different endometriums from different sites across different menstrual cycles and lots of donors. So I think in the future we will be able to answer this question better. I think, I mean, we cannot forget that this tissue, because of the cycling nature of the tissue, and the cycling is not always 28 days, it's approximate. So it's difficult to see the changes, because a lot of changes can be associated to the length of the cycle, and for this you really need a lot of donors to be able to answer this question.
I think I have another one from Anna Maria, I guess. What are the probabilities in the 2D projection plots? Did you try clustering in vitro and in vivo data together? Would different cell types still cluster together, or would there be a batch effect that would separate cells by their source?
Yeah, so the problem is that, I mean, we did try and they were not clustering together. And I'm not surprised, because the organoids have a more progenitor state, which is, it is what happens with the organoids. And actually, that's why it's important to compare in vitro and in vivo, because then you can push it forward to a more differentiated stage. So we tried to analyze it under the same manifold and didn't integrate that well. I guess, we just use, I mean, you can use the anchor from Seurat, that maybe there you will have a better integration as well. And that was, the probabilities were pretty high. It was pretty high. You can see them here. So they are quite high, all the probabilities. So we compare using logistic regression, but also we compare specific markers for each of the populations. So I agree that there are complementary ways where you can test or quantify these similarities.
So there's another one from our audience. Sorry if it was mentioned and I missed it, but does the proportion of cell types change throughout the cycle?
Yeah, it changed. That's a good question. So it changes. For example, the immune cells, we have an expansion of immune cells in the differentiation state compared to the proliferative one. So you have changes in the proportions and also changes on the identity. So it's both. So that's why, it's a challenging tissue to study.
So I guess those are all the questions I have found so far. Thank you again for an amazing talk, and sorry for the technical difficulties.
It's fine, thank you.
Thank you very much. And thank you again.
Thank you, a pleasure to present the work. Bye.
Bye-bye. So I guess it's now the time for a small presentation about NGS School Society. So this is my presentation that I will go into right now. Okay, so the reason we wanted to do this presentation is to tell you a bit more about what NGS School Society is. NGS School Society, NGS School is an idea, an organization, and something that was born into, something that started as an initiative to share the knowledge and train future adepts in bioinformatics and computational biology. But this is what it is right now, but it started just a summer school. And now that we are 4 years into, or actually we're starting a 5th year of our organization, more than 200 people from more than 200 countries were reached by our in-person events. However, that is, I hope now, much bigger, given that we are video recording all the talks and sharing them online. So yes, so NGS School is a community. It didn't start as it, but now that that we created those summer schools and that we created a way for people to meet and explore ideas and share them with one another, we also created a community. So before I'll want to tell you a little bit about the beginnings. So we wouldn't be talking about the beginnings of NGS School if we wouldn't mention Leszek. So 2016, Leszek first talked with Brona and Thomas at lunch about the training that he's been organizing in the International Institute of Molecular and Cell Biology in Warsaw. And there, then during this conversation, they started talking, and they decided that maybe it would be a good idea to organize a summer school. And that's how a lunch conversation turned into the first summer school that happened in 2016 in Slovakia. 2 years later, during the third school, we decided to formalize our organization and create an NGS School Society. And then we organized a 2019 school, and then we had huge hopes for 2020. However, we all know what happened. COVID happened, and we had to evolve with it. And NGS School and the whole world went virtually. So before we went virtually, we organized those schools in person. So as you can see, we had the school in Wysokie Tatry, Tatrańskie Czaj, and Statistics. So that was the very first school targeted at people making their first steps in bioinformatics. It was talking about the NGS analysis. The second school that happened in 2017, here titled as Stats Workout in the Gym, was actually in a single cell sequencing. The title was taken from the fact that because we had some mashup with the venue, we had to have one of our speakers, German Demidov, had to hold his talk in the gym. Then viewing Lublin through a nanopore, that was the school that we organized, and it was titled NGS School 2018, Nanopore Sequencing and Personalized Medicine, where we actually did some nanopore sequencing, which I'll tell you more about in a second. And then last year, the Adventures of Biostatistics in the Random Forest. This was the school about machine learning in biomedicine. So throughout the years that we organized the schools, we experimented, and quite literally during the nanopore sequencing and personalized medicine school, because we actually hosted a hackathon during which people, our participants, actually perform the experiments in the wet lab and then analyze the data. So what you can actually see on this picture here, the participants, who were mostly computational biologists, were holding the pipette for the very first time and generating samples. And one of those hackathons actually resulted in a publication that you can see here. Then next year, with 2019, for the very first time, we tried video recording, and we recorded 3 keynote speakers and actually streamed a debate that we held in the in the ethics of machine learning. In 2020, we stayed active. So we were quite prepared for the new school when Corona happened, and then decided to not give up and stay active. So we organized the tutorials and workshops. When we hosted a series of those tutorials under the name of NGS seminars. As you can see, we have 13 of them, ranging from introduction to programming in R and Python, and introduction to Linux, into also diving a little bit more into data analysis and programming in both R and Python. We had reproducible science with Nextflow, Jupyter Notebooks, and SnakeMake. And we also hosted Andrei with isoform detection from long-read sequencing. And of course, we did this conference, which you attended. Throughout our time, we collaborated with many amazing organizations, starting with the International Institute of Molecular and Cell Biology, Open Science Foundation. We hosted some training with RNA Club Warsaw. We collaborated with the Polish Association for Bioinformatics, Medical University of Lublin, Warsaw University, with whom we co-organized nuclear landscapes 2 years back, and with video lectures and so on. And we were generously supported by the Visegrad Fund, who actually sponsored all the NGS School, and hopefully will sponsor one in 2021, provided we will be able to organize one. But we also collaborated with Artigen, 7N, GenX1, the Company of Biologists, and Advanced Computing for Research. And what I'm getting at is that we did all that. We were able to organize those schools, and we were able to organize all those events because of the organizers and because of the people who dedicated the time and worked to do that. But since this is a little bit of a recruiting pitch, I'm going to tell you that while work is a huge part of what we're doing, we also have fun. As you can see here on this pictures, like you can see the working part, but you can also see the lower picture here. That's the fun part with Szymon actually having a drone. I don't know what you do with a drone, but guiding it to flying a drone and taking this photo with a drone. And I would want to give a huge shout out to the organizers of the Virtual NGS Symposium 2020. So I'll start with Karolina Sienkiewicz. She was the project coordinator and did a huge amount of work to get this all together. Same with Szymon Grabia and Urszula Smeczyńska, who were taking care of the IT bit, which was very challenging, but they did a lot of amazing work, and thank you for that. And I mentioned Anna Maria, Samantha, Gladys, Lydia, Natalia. They were all a great help in organizing this. So a huge shout out. So I'm going to end with telling you that if you want to get in touch, you can do that via our new website, that we redid this year. All done in R, so shout out to our stats community. You can shoot us an email at info@ngschool.eu or join us at any of the social platforms, social media platforms that I listed here. But what I'm going to say is that NGS School is it's a great community, and being part of the NGS School Society, you get to decide what we're doing next and shaping the next events we're doing. We're going to have a meeting very soon to summarize the past year and come up with the plans for the future. So if you want to be part of the organization, just let us know. Either write us an email, or, as I said, there are a lot of social media platforms that we're at, and you can always reach us there. And it will be a pleasure to welcome you to our organization. We're always looking for new people. And if you have any questions, I'm always happy to answer them whenever, whatever that may be. Thank you very much. And I guess with this, this is the end of our conference. It was such a pleasure to be with you for the past 2 days. Would I wish it to happen in real life? Probably, but this was great either way. And thank you for joining us. Bye.